Good morning from Nigeria. Greetings, ladies and gentlemen, conferees, and everyone present. It's a delight to welcome you to day 2 of the 4th International Virtual Scientific Conference of the Faculty of Pharmacy, University of Lagos, Nigeria. We welcome dignitaries seated, though virtually, and we want to take a recap of yesterday's program. Day 1 of the conference started with an opening message, welcome charge by the Vice Chancellor, University of Lagos, Professor Folashade Ogunshola, OON-FAS, ably represented by the Deputy Vice Chancellor, Development Services, Professor Afolabi Leshi. The keynote lecture was taken by renowned Professor Duncan Q.M. Craig, the Dean, Faculty of Science, University of Bath, United Kingdom. And he exposed the theme, Translational Research and Strategic Partnerships, Advancing Pharmacy and Health Systems. Before we go to the details of the summary of his speech, kindly permit us to take the national anthem, followed by the University of Lagos anthem. Thank you.
Nigeria, we hail thee, our own dear native land. Though tribe and tongue may differ, in brotherhood we stand. Nigerians old and proud to serve our sovereign fatherland.
once more for joining us. This is the Future Ready University of Lagos, the university of first choice and the nation's pride. We appreciate all the conferees that joined us yesterday, as well as our special guests. The keynote speech was taken by Professor Duncan Q.M. Craig, the Dean, Faculty of Science, University of Bath, United Kingdom. He spoke on the title, Translational Research and Strategic Partnerships, Advancing Pharmacy and Health Systems. He took us through the importance of translational research and doing cutting-edge innovative discoveries. It was a very detailed lecture, followed by questions and answers from conferees. It was very interactive. Thereafter, the first plenary speech was taken by Professor Afzal O. Rahman Mohammed. The title of his speech was Patient-Centered Innovative Drug Delivery Systems for Enhancing Healthcare Therapeutic Outcomes. He spoke mainly on pediatric medications with focus on orally disintegrating tablets. The speech was also very engaging. It showed innovation, and the end result was to make sure that the drugs have patient adherence. It was followed by a question and answer session from the conferees, and it was also a very stimulating and informative speech. The second plenary lecture was taken by Professor Amala Sum Yahanav. Professor Amala Sum Yahanav is the director, Benfra Botanical Dietary Supplements Research Center, Oregon Health and Science, University of Portland, Oregon, United States of America. And she spoke on translating natural products into therapeutic innovations and advancing phytomedicine from bench to bedside. She let us to know that translational research it's important for collaborations, strategic partnerships, it will enhance health systems and advance pharmacy. Day 2 of this conference will be having 3 speakers and also breakout sessions for the parallel sessions. The one breakout session, we took over 40 abstracts for day 1 for poster presentation in the different departments: clinical pharmacy and biopharmacy, pharmaceutical chemistry, pharmaceutical microbiology, pharmaceutics and biotech, pharmacognosy were the different breakout sessions for the parallel session. And today, our keynote speaker, our third plenary speaker, who will be the first speaker for today, is Dr. Fidel Ntiekang from medical chemistry. He's speaking on medicinal chemistry, which means translational research, innovations in drug design and delivery. He is speaking from University of Bath, Cameroon. He'll be joining us soon. Boya. Our first speaker for today, which is the third plenary speech for this conference, is Dr. Fidel Ntiekang from University of Boya, Cameroon. His speech is on medicinal chemistry meets translational research, innovations in drug design, synthesis, and delivery. And to read his citation is Dr. Bamishaye Oyewaluja. So you turn your camera on, Prof, while your section is being read. Thank you. Dr. Oyewaluja, please.
Good morning once again from the University of Lagos. I will be reading the citation of Dr. Fidele Ntiekang. Dr. Fidele Ntiekang is an associate professor of pharmaceutical chemistry at the Department of Chemistry, University of Boya in Cameroon, where he also heads the Center for Drug Discovery. After studying chemistry and physics at the University of Douala in Cameroon, he completed his PhD in physical sciences at the Center for Atomic Molecular Physics and Quantum Optics at the University of Douala, focusing on computer-aided drug design. After working briefly at the Department of Chemistry at the University of Boya, he moved to Germany, where he carried out his postdoctoral research under Professor Wolfgang Spiel, leading to habilitation in pharmaceutical chemistry. His research has been focused on chemoinformatics for natural product research, a topic in which he is an author of a book series. Besides, his team has been leading and building and testing small molecules of natural and synthetic origin in the search for active molecules, such as antiviral, anti-malaria, anti-diabetic, and anti-cancer agents. He holds several editorial board appointments and is passionate about drug discovery from natural products identified from African medicinal plants, as well as pseudo-natural products derived by fragment-based hybridization. He has received several awards, including the Commonwealth Professional Fellowship at Leeds in UK, George Forster Postdoctoral Fellowship from the Alexander von Humboldt Foundation at Bonn in Germany, the Carlistos Juma Science Leadership Fellowship from the Bill and Melinda Gates Foundation, which was $1 million, and another joint grant award by the BMGF and LifeHack to develop an African-derived natural product box. He is a part of several consortia, including being a founding member of the Grand Challenge African Drug Discovery Accelerator Consortium, a founding member of the Cameroon Academy of Young Scientists. He has previously been a guest professor at the Technische Universität in Dresden, Germany, and an adjunct professor at Nelson Mandela African Institution of Science and Technology in Arusha, Tanzania. His major contribution in the development of African Natural Product Database. He is the 2025 Royal Society of Chemistry Natural Product Reports Emerging Investigator Leadership Award winner, and in fact, the first African to receive the award, and also a member of the first cohort of the Cameroon Academy of Young Scientists. So, conference attendees, please help me to make a resounding welcome for Dr. Fidele Ntiekang, who is going to take us through a delve into his research in chemoinformatics and drug discovery. Thank you very much.
Thank you very much. I'm so delighted by the presentation. I hope you hear me.
Yes, we can hear you loud and clear. Please extend conferees. The title still stands. I want to let you know that Associate Professor Ntiakeng wants to intimate us of what has been happening in his lab. So the focus of his lecture this morning will be on unlocking the continent's drug discovery potential through computational applications. The floor is yours, sir.
Thank you very much. I hope the slides are moving. The slides moving.
The slide is moving, sir.
Perfect. All right. So thank you very much for the presentation. I'm really honored to be part of this big conference. Actually, I would say that I'm your neighbor. I'm just next to you. Actually, I don't need a visa to travel to Lagos. I don't need a visa to travel to Nigeria. So I have a long history with Nigeria. Actually, many of my colleagues are Nigerians. I went to Nigeria for the first time. I was not 20 years old. And right now, I am also employing a postdoc from the University of Nigeria in Nsukka. And also, one of my first PhDs was from Nsukka. Actually, he's now a lecturer back in UNN. So I have a strong connections and collaborations with Nigeria. Now, starting from there, I would say that today's talk is unlocking the continent's potential for drug discovery through computational applications. The idea is that Africa is a very huge continent, right? And so being a very huge continent with huge diversity in terms of populations and in terms of plant species and so on, we also have a huge diversity in terms of chemical composition of the plants that grow in Africa. So this is a strength we have. Of course, we also have another strength, which looks like a weakness, and the strength is that Africa carries 26% of the disease burden of the world. So if we have the plants which have been providing solutions for our healthcare needs, and we also have the diseases, that means that we have what it takes to discover the diseases, the drugs that tackle the diseases that the African populations are suffering from. So today, I would show you how diverse our natural product is, and then we will now show you some of the challenges that we have faced in drug discovery, beginning from medicinal plants, and we see where chemoinformatics, which is simply chemistry with the use of computers, comes in to solve some of the problems that we face. And the second thing is we will show you a couple of case studies where we'll show you how we have been able to take it from natural products to assays. We show you also how we take it from natural products to through chemoinformatics to get pseudo-natural products which have been confirmed to be active. And then, of course, we will now be able to propose a couple of direction points that we think is a way forward towards developing potential drugs from natural products beginning in Africa. Now, the first thing we see is that Africa is very diverse. As I mentioned before, we have more than 50,000 species growing in Africa, and some of them are endemic to Africa. And historically, we know that many of the drugs that are used today, that are in the market today, including artemisinin and Taxol and quinine, have had their origin in plants. Now, the question that everybody asks is, when are we going to have a drug in the market that comes from African medicinal plants? Because we know, of course, that artemisinin came from Asia and quinine came from Latin America, okay, and Taxol came from North America. Now, the question we have been asking is, when is Africa going to put a drug in the market? Having this diversity that we have mentioned. Beside that, we know that from WHO reports, about 80% of the populations of Africa depend on traditional use of plants directly or indirectly for their healthcare needs. So with this in mind, you would quickly realize that the first challenge that we face as Africans to transform our resources, our natural resources, our biodiversity from plants into drugs face two major challenges. The first of them being limited funding. So often, African scientists are reduced to sample collectors. In essence, you collect your samples and you jump on a flight and you try to spend some time abroad where you have access to high performance equipment like NMR and so on. Then at the end of the day of your stay, we probably have identified one or two bioactive compounds. Of course, you are excited and you get a publication out of it. And after 3, 4, 5, 6, 7, 8, 9, 10 visits, you have what it takes to become a professor. Of course, then when you become a professor, what's the next thing? Is that you start campaigning to become the vice chancellor. So in essence, the challenges we have in Africa have been around the continent are the same. Limited funding, limited infrastructure. So with this, how do we get around it? One of the ways of getting around it is the use of computers, chemoinformatics. The reason being that computers will make the task to go faster. Now, today, you don't really need to have all the sophisticated equipment because now you have cloud computing. Today, you don't really need to have a huge expertise in terms of the background of computer-aided molecular design, because a lot of tools which take all the theories and the concepts and feed them into one, and some of them are available as web servers, some of them are available as packages that are standalone packages. So in essence, with the use of computers, you can potentially screen several millions of compounds as compared to wet lab screens. First of all, for wet lab screens, you need to have access to the reagents, you need to have access to the instrumentation and so on. With computer-aided screens, which we usually call virtual screens, you don't need all of that infrastructure. All you need is the molecular files, which you can actually, most software can run several millions of them. So you are interested in finding a lead compound, for example, then you have several millions of compounds in a database, which you can either use artificial intelligence methods, or you can combine these together with structure-based drug design methods. That means if you have a chemical structure of the drug target, either in the form of an X-ray structure from the protein data bank, or in the form of a homology model or something like this, or you have an active compound which has, for example, some side effects, and you want to find analogs that contain the same pharmacophore, you want to probably use ligand-based methods and so on. So there are several methods that can help you stream down the number of compounds you want to screen. So in essence, you don't need to screen several thousand compounds. What you need is you can start from several million compounds in the form of a database, that molecular files that you carry out a simulation, and you narrow down the number of compounds that you are eventually going to assay. So you may start with 1 million and only assay 15 compounds. So which means the cost of screening becomes less. So you don't need a high throughput screening facility. The cost of screening becomes less because you eventually screen just a few number of compounds from your database, which fit a certain number of criteria that you have established as filters from the start of your virtual screen. So with that, the second advantage we have is going to be the sustainable development of our resources, because in essence, the way natural products research was being done before was simply you go to the forest and harvest this plant, or you go to the village, talk with some traditional healers, they give you the species that they use to treat a particular ailment. Of course, you prepare extracts which typically are not prepared the way the traditional healers do it. You come to a laboratory and can use chemical solvents to extract, to carry area extraction. Then you use chromatography to separate your fractions, and eventually try to see if it's possible to crystallize out active compounds from there. Of course, then when you carry out your assay, sometimes the activity is not what you expected, because sometimes the compounds just act in a synergistic manner. But then if everybody is doing that, the risk we have is that we are going to bring down our forests, because that's the diversity that we have as our heritage from our ancestors. So if everybody goes to the forest, I live at the foot of Mount Cameroon. If everybody goes to Mount Cameroon and harvests these plants, first of all, the plants may become extinct. Secondly, this way is not sustainable. So in essence, if you use computers, you are going to narrow down to a few compounds which you can, if you are working with databases that's well annotated, you can now be able to trace back these compounds from the plants that made them. So you go and harvest just those plants. The second advantage you have is that eventually, when we have a compound that's really, really active, then the sustainable development of these plants might require going through Nagoya conventions where the two countries, for example, you are working with a partnership with a foreign country, the two parties concerned benefit. So win-win situations are going to be there if you can be able to trace exactly the source of the species and go for it. Now, what we have been doing for the last 10 to 15 years in our group is that we have been cataloging natural compounds that were identified in African medicinal plants. So what have we been doing? We simply do what? We simply go to the literature, and when we go to the literature, we simply find a number of give me a moment. Yes, thank you. Yes, we simply find articles and theses, for example, that were published, and then we check in there country by country to be sure that these species were harvested in Africa. Then, of course, we eventually retain those that were harvested in Africa, and we collect this information in form of tools, because we want to build an SQL database. So in essence, these are relational databases. So we want to be sure that we know all the molecules that were identified. We have the names of the species that were identified, the identification of the species that goes through, for example, the taxon IDs. We want to be sure that the references are correct, because if someone is interested in this compound, they can easily go back to the reference. They will see how the procedure of isolation of the compounds and how the compound was characterized. Right now, actually include even the spectral data of the compound. Then, of course, the references include also the person that work on it, so that in the event where you are interested in a particular compound, you can actually contact the person who worked there if they have samples for screening. Then after collecting all this data, we make the structures, the chemical structures, into files, which we are going to from there. There are different file formats, like SD format, like multiple formats, and so on. From there, we extract what we call the SMILES, which are simple molecular line entries, which in informatics is used to be able to identify the chemical structure in a simple manner. Today, there are a lot of software that can transform the SMILES into chemical structures, which you can visualize on the computer. From there, we are able to generate from the SMILES, calculate the physical chemical properties and the toxicity properties, related properties, and all of these have been in one database which you can have access. If you go to african-compounds.org, you can actually go there and get all this information. Right now, as I speak, we have more than 11,000 compounds in there. We have more than 1,700 species, and right now we are still updating again. So hopefully, before the end of the year, we'll probably talk about like 15,000 compounds which have been identified from different regions in Africa. Okay. Now, but in this whole journey, there are several challenges. And among the challenges is having access to the physical samples of the compounds. Among the challenges is identifying the species in a specific way. Sometimes there are errors in identification of the species. Another challenge, of course, is the use of computer tools to be able to, of course, sometimes the students are tempted to think that the computer tools are video games that they play with, but often there's a lot of knowledge behind that you need to understand and how to capture and how to interpret your data. So all these challenges that we face, often we summarize all of them in this RSC paper that was published last year, or 2 years ago, sorry. And then let me talk briefly about visual screening. Now, in visual screening, you don't actually need to have a sample of your compound to be able to enter the laboratory and carry out your assay. In visual screening, what you need is a molecular structure which should be well annotated. It should be the chemical structure should be correct. Then you simply use computer methods which you are going to use to either identify which target this compound can bind to. That's called target fishing. Or if you have a compound that is bound to a target and you are not too happy with the compound, either for some other reason, maybe the affinity is not too strong. Sometimes the fear is that there are some side effects related to this compound. You may want to change the chemical structure. That say you want to modify the structure, but that should bind to the same target. That is called scaffold hopping. Now, of course, another way to do it is to identify the bioactive pharmacophore features of your compound that are responsible for the activity, so that you can now find other compounds that have the same features which can bind to the target. Okay, so all these are approaches that we use. Often nowadays, artificial intelligence has come to make things even go faster. So in essence, I'll give you some examples that when we made in 2015, we published this database of 1,000 compounds from Africa with at least one activity. It was called the AlphaDB. This paper has been so highly cited. Actually, I got my Humboldt Fellowship from it. My former Dr. Babiaka got a Humboldt Fellowship. My colleague, Denis Zofu, was second author, actually won an award at Young Scientist Africa, and so on and so forth. So, using visual screening of this library, had been shown that this compound you have a Medicinal, which was in that database, had some activity against Ebola. just by visual screening and experimental assays, they found activity 1.3 micromolar against Ebola. I'll give you another example from the Pan-African Natural Product Database, which we made sometime in 2014. We were able to also carry out pharmacophore-based visual screening, and we were able to find baldine, this apophene alkaloid, have activity against HIV. In essence, by blocking the ion channel, what we call the VPU channel of HIV. So in essence, computer-based methods can make things go really fast. But if I had to carry out the 1,000 compound, which I didn't have access to, all of them, and I say all of them in against VPU, then it would take me like 3 to 4 years, and it would take a lot of money to carry out this assay. So in essence, when we carry out our visual screen, we started off with about 500 compounds from the Pineapple library. We ended up with about 10 compounds, which we suggested as virtual hits, which were acid, and baldine and another active compound were both active in the assay. So this is how things can go really fast by use of computer resources, cutting down the cost. So right now, when we got the Kalisto Juma Science Leadership Fellowship from the Gates Foundation, it was $1 billion. Actually, it was a call upon invitation. So we were about 14 of us that were successful, including 2 Nigerian professors, Professor Iruka Okeke, that some of you may know, and also Professor Ife Anebo, whom some of you may know. I'm talking to a Nigerian audience, I suppose. And so the idea, our idea of our project was to build around this idea that we call nature-inspired discovery of novel antivirals. So what was it all about? The first thing, we would inspire ourselves from the natural compound databases from Africa. Then we would look for chemical structures from plants. And then we, of course, have an in-house library of natural product mimics. That means we also work with compounds that look like natural products, but they are not natural products. So in essence, we build them from fragments of natural products. So we call the natural product mimics. So we use visual screening, both machine learning methods and physics-based methods, including docking and molecular dynamics, etc. We combine these together, and we're able to identify compounds which target HIV by reversing latency in HIV, or compounds actually that bind at the time the big story was SARS-CoV-2. So at the time, coronavirus was a main issue. So we were interested in the main protease. We were interested in compounds that block the fusion of the spike protein and the human angiotensin 2 conversion enzyme, which was important for binding and transmission of the virus. Now, so the approach we're using is, there are several approaches. I'll introduce you to one of them approaches, which is called fragment-based drug design. So what do we do in principle? In fragment-based drug design, what you need is a library of fragments. That means you're interested in compounds that are small, typically molecular weight less than 250 Daltons, preferably. Sometimes molecular weight less than 300, that's fine. So we are interested in compounds that are small. That means that the fragments are going to be recombined. So in essence, you have a drug target that can be protein, that can be, for example, a crystal structure of protein you got from the PDB, and you are interested in knowing which of these fragments can potentially combine to form a compound that has activity against your target. In essence, what you need is not the compounds to dock in the binding site like you typically do. What you need are the fragments. So in essence, you need to make sure you have a fragment library. Now, once you have a fragment library and you dock them into the binding site, now see in which ways can I combine the fragments to make whole molecules, which I will now have to synthesize and assay. So in essence, after you have combined the fragments, you have a complete compound. This compound can now be tested in the assay. So this is the way we do it. But of course, the challenge you have to have is to have fragments that contain the pharmacophores that will bind to your target. So in essence, for our work, since we know natural compounds are very huge, most natural compounds don't fit the criteria of fragment likeness, we're obliged to go around that, and I'll show you in a moment how we do that. And so how did we do that? First thing we did at the time is that we are not yet updated we don't have yet complete update of the African Natural Product Library. So when we started off the project, we started with NANTP, which is the Northern African Natural Product Database, which we had made at the time, and together with the East African Natural Product Database, which together was like 6,500 compounds. We also took PubMed, which was a Congo Basin medicinal plants natural product database, which we did sometime and published in the RSA Advances, contained about 3,200 compounds. We added to that AfroDB, which was about 1,000 compounds at a time. So when we put this together, we had more than 10,000 compounds, which are unique compounds. So we removed duplicates, of course, we know some compounds are found in several databases. Then from there, the next step was to do what we call fragment generation. That means all these compounds, you see how huge they are. If you combine two of them, you simply not be able to handle that from a medicinal chemistry point of view. So we actually had to, in these compounds, that we take a compound, you split in the fragments, such that the fragments respect the criterion of fragment likeness. And the procedure that is often done is called recap. That simply means retrosynthetic recombination of fragments. So in essence, you go compound and go backward and tell yourself, if this compound was made, which were the fragments that were combined to get, that were hypothetically combined. So there are a number of rules that are implemented. You implemented recap in MOE, and then we got these fragments. Then the next thing is we saw that we had about 5,277 fragments. Now, when you have fragments like that, you do not, you are not filtered out the compounds that really fit the criterion of fragment likeness. So when we did that, we had less than 1,000. It was nice to handle. So with less than 1,000, we're now able to just the fragments, and the fragments were what was docked in the binding site. In this case, we're dealing with an enzyme that is called the human histone deacetylase 1, which is one of the mechanisms of HIV reversal. Is that when HIV cells are dormant, HIV-infected cells are dormant, one of the ways they can be reactivated by latency reversal, one of the mechanisms passes through histone deacetylase inhibition. So what we did here is we simply got a structure from the extra structure from PB, which we prepared, and of course all our fragments were docked, as I mentioned in my previous slide. We docked all of them, and then we try using Schrodinger. Of course, we now rank them by scoring them. Then eventually, now when we carry out the scoring, we want to see which of the fragments are having the highest affinity. And then we now see how do we, looking at the binding site, the way the compounds will display in the binding site, each fragment will look for its own position. And we see how do we recombine them within the binding site to get compounds that we can synthesize. So this is the approach. So we have several where you dock, you rank it, you have several fragments that rank. So you now begin to check them, how each of them fits in the binding site. So you are going to have something like this. It's going to be something like several of them, several fragments. Each fragment will look for a particular preference to a particular section of the binding site. Now you are going to overlay them and see which of the fragments on the left-hand side should I combine with the fragment on the right-hand side to get a compound, a complete compound. So you have something like this. In this case, we had an indole here, and you see we combined an indole with a small fragment. In this case, we had several possibilities. For example, we have here together with an indole, they can give this possibility of having this scaffold. Now we also have, of course, then we have here a carboxy compound together with this indole. What can we get? So we had several suggestions in terms of which are the fragments were there, which were the fragments that docked best. We now try to combine them theoretically. So if you combine this and this, you're going to have this on, of course, this is just on the computer. You are not too sure if you can synthesize them. So we have 3 scaffolds, which we designed only using a computer. And now we went back to the lab and see how do we synthesize these compounds. The first challenge we had was combining the ketone and the indole, because several reactions were attempted and all of them failed. Eventually, we realized that the reason why, of course, starting for example, one of the reasons why this reaction may have failed was because we had a very strong base here. We tried here, maybe we replace it with ethanol. What happened at the end? It didn't succeed. Just we used solvent-free, and then just by heating, it failed. But we realized that the way of synthesizing this compound was by initial protection, a bulk protection, because the NH group here was literally in competition with the solvent. So in essence, when we use bulk protection, using the same reagents as before. The compounds was formed in one step, one easy step, without the use of one simple step with the use of bulk protection. So the bulk was deprotonated immediately, and the compound was formed in one step. And of course, this new approach, it was the first time that indoor chalcone, because combination of this ketone and the indoor form indoor chalcone, it was the first time that we're able to describe the synthesis of indoor chalcones using this approach. So the idea, what I was inspired was the combination of fragments. Of course, this paper was published last year. My PhD student did this work in the lab. And beyond that, we have done several other tasks, including since the screening of both pseudo-natural compounds and natural compounds for the ability to block the fusion of the virus protein from the spike protein from coronavirus to the human angiotensin II converting enzyme. And we have some of these recently published. And of course, beyond that, I'll show you here this very, very potent compound here that we carry out both the synthesis and the biological assay and the computational work, able to show how harmonious this whole approach can go to. So some of this work was done by master students actually in our lab. And in this work, we were able to show, for example, that this compound, which I didn't show the structure here, actually is potent against blocking the spike ACE2 interaction at 160 nanomolar concentration. And of course, the good thing is the compound was also very selective towards this protein-protein interaction because actually we know that spike ACE2 is a protein-protein interaction. So we a random interaction in humans and saw that this compound actually was really selective as well. So we saw that the compound was active against also all the wild type, that's the Wuhan strain, the Delta, the Omicron, and the Lambda for both the selectivity was very clear. So in essence, we showed here that the compound was binding actually to the interface, not really like the target initially was the interface between the receptor binding domain of the viral spike and the human ACE2, but in essence we actually saw that the interaction was taking place in the angiotensin II side, which is a site that we have been exploring actually in recent works in our lab. Now, when I look in the future, what I see is that such works can be possible in Africa through collaboration and capacity building. So in essence, one of my big focus, I'll be turning 50 next year. So one of my big focus now is how do I pass on the torch? So in essence, my focus is working with students, training them, and building a research team. So we are looking at also the use of artificial intelligence and machine learning as one of the ways forward towards enhancing drug discovery on the continent. And of course, I also look at the sustainable and the ethical management of plant resources in Africa. Otherwise, we are going to lose our plants in the next decade. So in next generation, sorry. So in essence, one of the ways I'm looking at is how do we capture the genetic resources and how do we keep them in a digital format that if plants go extinct, what becomes of our next generation of scientists and all of that. Then I'll show you a couple of pictures here. As I mentioned, our secret collaboration here, my collaborator from Bedside Institute in Philadelphia, my collaborators from Spain and all of that. So we often send our students out there for training. Sometimes colleagues come over for training and so on. And then, of course, several pictures of our drug discovery center that was recently built from the grant. And so, in conclusion, I would say Africa's natural products represent an incredible and untapped resource for drug discovery, and the integration of chemoinformatics, the traditional uses of plants can be very, very crucial to drugs for the next generation. This is only possible through collaborations. I will not end without acknowledging the funders, particularly the Gates Foundation, for giving us Juma Fellowship and the group that has been very instrumental to our work alone. It's quite a huge group behind all this work. So thank you very much and looking forward to your questions.
A round of applause for Prof. He is the head, the Center for Drug Discovery in his department and associate professor of pharmaceutical chemistry You all can agree with me that it was a stimulating, impactful, and brilliant lecture. Another round of applause, please. Yes, Prof let us to know the importance of chemoinformatics in drug innovation, and he put us through the works that have been done in his lab, cutting-edge research, and the publications. Before we take the Q&A, I would like you to know that he was so magnanimous to send us the link to the African Natural Product Database that has been used to catalog the structures of bioactive compounds derived from African natural products. He let us to know that if we do not save our biodiversity, we are going to lose our medicinal plants 10 years from now. So we have to go into genetics and molecular studies to digitize what we have. And that is the essence of this conference. And he encouraged us to collaborate and so that we can do cutting-edge research. Please, we have a few questions in the Q&A question box. I want to read. Oluwa Shefumi Ajose says, good morning, sir. Thank you very much for your well-explained presentation. My question is, as a recently graduated pharmacist, how can I register to join one of your research teams?
Okay. Yeah, it's a question I get from everywhere. The first thing is, we are the second center for drug discovery in the continent. So we have been inspiring ourselves from the H3D Foundation, H3D Drug Discovery Center in Cape Town, South Africa, headed by Professor Kelly Chibale. So H3D focuses mostly on postdocs. So in essence, they have the Department of Chemistry where Professor Chibale is a professor of organic chemistry, where they train masters and PhD students. But then they have another drug discovery center where they actually work mostly with postdocs so that they do real, the idea is to get products to the market. Now, in our Center for Drug Discovery, I see work in the chemistry department where I train students in pharmaceutical chemistry. But the Drug Discovery Center is apart and it's mostly from external funding. So what I usually do is that my fishing pond is the chemistry department. So when I teach my students at the master's level, then I identify the very brilliant ones that I can really focus. Then I engage them now in projects. So in essence, if you are really interested in working with us, the first thing would be to register for a master's. Then when you register for the master's and you are, say for example, you're doing your coursework, we can now offer you something like an internship where you can actually do your hands-on in the lab. So that's the way we do it. But that would just grab a student because we are not necessarily running master's program in drug discovery. We are planning it. We are not doing it.
Thank you so much for that answer. That is the only question we have. And it's now time to present.
Some people are asking for time for my contacts. I think they're on the slides. The contact and the link to the database, they're all on the slides. I share the slides with the organizers.
So the contacts and the link to the database.
Yeah. They're on the slides. I'll share the slides with the organizers.
So much. It's now time to present to you with. Thank you, sir. We'll share the slide as he has shared with us with the registered participants. Kindly register. We call on Professor Chukwuemeka Azubike to present the award to our speaker, Prof. Sir.
Thank you very much for the impactful lecture. On behalf of our Faculty of Pharmacy, University of Lagos, and the 4th edition of the Faculty Scientific Conference, I am glad to present to you Dr. Fidel Ntikang as the speaker at this 4th Faculty of Pharmacy Virtual Conference on this day, 11th day of September 2025. Thank you very much, sir. God bless you, sir. We will send it to you, sir. You will get it
All right.
soon. Thank you very much.
Okay.
Thank you so much, Professor Chukwemeka Azubike, for the presentation of our word. It's now time for the fourth plenary lecture. You all can agree with me that it's really an insightful conference because we are going to collaborate, engage, and continue interacting. Now, yes, thank you. To read the citation of our fourth plenary speaker, in the person of Professor Nneka Regina Agbakoba. She will be speaking on harnessing the microbiome for antimicrobial stewardship and resistance mitigation. Harnessing the microbiome for antimicrobial stewardship and resistance mitigation. Professor Nneka Regina Agbakoba. And to read the citation is Pharmacist Chijioke Ezobiora. Thank you.
Thank you very much, Ma. Good day, esteemed participants. Please permit me to proceed according to the already established protocols. My name is Ezobiora Chijioke Emmanuel, and I have the privilege to read the citation of Professor Nneka Regina Agbakoba. Prof. Ma, kindly turn on your video as I read your citation. Professor Nneka Regina Agbakoba is a professor of medical microbiology in the Department of Medical Microbiology and Public Health, Faculty of Medical Laboratory Science, Nnamdi Azikiwe University, Awka, Nigeria. She had her BSc in microbiology from the University of Ibadan, master's degree in medical microbiology from the University of Nigeria, Nsukka, and doctorate degree from the University of Ibadan, Nigeria. She is a fellow of both the Medical Laboratory Science Council of Nigeria and the West African Postgraduate College of Medical Laboratory Science. Administratively, she has been a two-time head of her department and was the Dean of the Faculty of Health Science and Technology from 2012 to 2014. She has also been the Deputy Provost and the Acting Provost of the College of Health Sciences of Nnamdi Azikiwe University, Nnewi Campus, between 2018 to 2023. Currently, she is the coordinator of the Molecular Research Laboratory at UNIZIK, Nnewi Campus. She was the pioneer treasurer and later the registrar of the Nigerian chapter of the West African Postgraduate College of Medical Laboratory Science. Professor Agbakoba has been a great mentor to many students at undergraduate, postgraduate, and fellowship levels. On research interests, she has worked extensively in many areas of medical microbiology, especially in the field of mycoplasmology, human microbiome, metagenomics, and human probiotics. She has published widely in both local and international scholarly journals and has over 100 publications, 4 book chapters, and a textbook. Some national and international organizations she belongs to include American Society of Microbiology, International Organization for Mycoplasmology, Association of Medical Laboratory Scientists of Nigeria, to mention but a few. Professor Abakoba is happily married with children. Ladies and gentlemen, please join me in a warm welcome to Professor Nneka Abakoba as she takes the podium. Thank you.
Prof Ma, if your bandwidth is low, you can turn off your video while you share your screen. If your bandwidth is low.
Can you see it now? Hello?
It's coming up, Prof.
All right, all right.
On slideshow.
On slideshow, okay.
Thank you, Prof.
Good morning, everybody. Am I being heard?
Loud and clear.
Hello? All right, thank you.
Thank you,
Good morning once again, everybody. I'm highly privileged to be and honored to be in your midst this morning. Thank you for inviting me to come and be a speaker here today. So I'm here to talk to you on the topic, harnessing the microbiome for antimicrobial stewardship and resistance mitigation. I'm going to be talking to you under the following outline here listed. By way of introduction, antimicrobials are agents or substances that act against microorganisms, be they bacteria, fungi, parasites, viruses. Antimicrobials are usually derived from different sources such as microorganisms, plants, animals, synthetic or semi-synthetic materials. For antimicrobials to work effectively, there had to be a good antimicrobial stewardship program. And what is antimicrobial stewardship program? It refers to a coordinated set of actions designed to promote responsible use of antimicrobials, and its overall goals include ensuring optimal patient outcome while minimizing the emergence of antimicrobial resistance and reducing harmful side effects to patients and also controlling healthcare costs. Strategies have to be involved for good antimicrobial stewardship. Some of these strategies include to improve the selection, dosage, duration, and route of administration of antimicrobials. Infections usually are still being treated with antimicrobials. Wow, what's happening? Hello? Wow.
Your network, Prof. We can no longer hear you.
Okay. Hey.
Okay, you are back.
Are you hearing me now?
Yes, Prof.
Hello? Hello?
Yes, please. We can hear you, Prof.
Sorry. All right. Okay, I was saying that before the advent of antimicrobials, the rate of death by infectious agents was very, very high. And infections now can be treated well with antimicrobial agents. But then, before you know what is happening, these microorganisms began to develop resistance to some of the drugs, especially bacteria. No matter what you do, bacteria will always find a way, not a way, just ways to circumvent these drugs and become resistant to them. Soon, phrases like drug resistance emerged. Much later, another phrase called multiple drug resistance followed suit. And now we have the extensively drug resistance. We could name MDR. All these are because microorganisms are trying to find ways to evolve and then dodge the work of the antimicrobials. They do what? They become resistant to them, become totally different organisms that were earlier susceptible to the same drugs they are now resistant to. Aside from resistance, antibiotics also cause a lot of disturbances, which we call dysbiosis in the microbiota of humans. And such dysbiosis can lead to disease conditions like antimicrobial associated colitis, infectious diarrhea.
Can you hear me, Prof? Your slide is not moving, please. Thank you.
Are you hearing me?
We can share for you. Your slide is not moving. We can hear your voice, but your slide is not moving. Do you want us to help you share your slide from here? To move it?
You can, if you share it from there, can I move it?
You tell us when to change it. Yes, we can turn it for you. We can change the slide for you.
Okay. Okay. Go ahead.
We are just seeing your title slide. Yes, you can share from here.
So which slides are you now on? Are you So please go down. I'm already at go down to maybe slide number 5 or 6.
Slide number.
I think I'm down. Is that slide number 5?
Number 5. Slide number 5.
Go back one more slide. Go back again. No. You're going down. Go up.
Forward.
Fortunately, I didn't number. Move back. No, I've not reached microbiome. I'm still introducing the topic. Go back.
Slide 5.
Go back to 3. Let's see.
Slide 3. Introduction. We can start from introduction. Is that fine, Prof? This is slide 3. We can hear your voice, please. Can you hear us, Prof? We can't hear you, Prof. Our Prof is back. Kindly unmute and speak, Prof.
Yeah. Are you hearing now?
Yes, please. Thank you.
Can you hear me now?
Yes, Prof.
Oh, let me just be fast about it. The network, I've been moving around. The network is so unstable today. I wonder why. So I was saying that the antimicrobials are actually substances that act against any type of microorganism, and they can be derived from different sources. Please go on to the next slide. Move on to the next slide. So for effective antimicrobial, there has to be a good antimicrobial stewardship program on ground. And the overall goal is to ensure optimal patient outcome while minimizing the emergence of antimicrobial resistance, reducing harmful effects to patients, and of course, controlling healthcare costs. So strategies involved is to improve the selection, the dosage, the duration, and routine and route of administration of antimicrobials. Next slide. So this antimicrobial resistance has continuously risen, and this is due to the use and overuse and misuse of antibiotics. This rise, of course, presents a huge challenge to public health, to undermine the efficacy of current drugs, antimicrobial drugs, and compromises the management of infectious diseases. Next, please. Globally, antimicrobial resistance has become a major and escalating global health threats. The rise of multiple resistance has led to increasingly recurrent and difficult to treat infections, as well as prolonged hospitalization, resulting in higher morbidity, mortality, and high healthcare costs. Next, please. Let me just. So antimicrobial resistance has significant economic costs. The World Bank has estimated that antimicrobial resistance could result in $1 trillion additional healthcare costs by the year 2050, as well as 1 to 3.4 trillion gross domestic product losses per year by the year 2030. Next slide. Next slide. The scarcity, the problem is that this scarcity of effective therapeutic options, there are not much therapeutic options. And this does what? It exacerbates the crisis. And hence, there is need, urgent need for innovative solutions as far as taking care of resistant infections are concerned. And this quest to deal with antimicrobial resistance has actually led to explore the role the microbiome will play in mitigating such resistant infections. Now, what is microbiome? Next slide, please. The microbiome is a community of microorganisms and their genes that live in specific environments such as human body, where they interact with each other and their environments, forming complex ecosystems. Microbiomes are intricately balanced ecosystems of microorganisms at various body sites that play pivotal roles in human health and disease. Next slide. So the human body, the human microbiome includes trillions of bacteria, fungi, and viruses that reside on and within the human body. And the human body has several habitats, or what we call niches. These are small areas in the body. And each of these habitats or niche plays a unique, harbors a unique microbiome composition. The diversity of microbes normally in each microbiome varies from habitat to habitat. Next slide, please. The composition of human microbiome, of course, differs from person to person. What is in this person's body will not be what is in another person's body. It differs, and each of them has several roles to play. Some microbiota are known to be predominantly present in some habitats, where they most often play protective roles in identifying and destroying invading microbes that come into their habitats. In the work we did, we reported that firmicutes, followed by proteobacteria, actinobacteria, bacteroides, and fusobacteria, dominated the oral microbiome of the subjects we studied. Likewise, there are other habitats like the guts, the skin, the genital tract, the nasal and ocular. They also have their peculiar microbiome, playing various protective roles. Next. Some critical roles played by microbiome in the body. They include the food digestion, detoxifying chemicals in the environment, barrier functions as first line of defense. They prevent the growth of harmful microorganisms and disease progression. And they also help the beneficial microorganism to colonize the habitats. Next, please. Microbiome in antimicrobial stewardship. You know that the gut is a very essential part of the microbiome, the gut microbiome, because most drugs that is taken orally. Apart from few injectables, most antimicrobial drugs are taken orally. And when they get to the gut, they do a lot of things. So there are key stewardship strategies that should be borne in mind when prescribing antimicrobial agents for sick patients. So some of these include using narrow spectrum agents. The prescriber should, as much as possible, avoid broad spectrum agents as much as possible, because when such gets into the gut, in trying to take care of the pathogens, they also do away with the beneficial microbes in the system. So using narrow spectrum agents, minimizing antibiotic exposure. If it can be avoided, it should be avoided or at least minimized. Then also, the drug developers should develop drugs that reduce the gut microbiota disruption. Next, please. Next. Other things to be borne in mind is when the patient is recovering, researchers should be able to pick out the biomarkers that may help individualize antibiotic regimens, lowering unintended antimicrobial exposure to the gut microbiome. So stewardship programs increasingly advocate for preserving healthy microbiota as part of responsible antibiotic use, so as to prevent downstream risks. Microbiome-driven strategies to combat resistance. So the microbiome should leverage on the the body should leverage on the resident microbial community to suppress pathogens. It should enhance host defenses, reduce the need for broad spectrum antibiotics, I've said it before, and evidences are coming up that indicate that manipulating microbiome can influence pathogen colonization, inflammation, and gene flow related to resistance. Next. So there are mechanisms that microbiota use in combating MDR infection. One of them is the nutritional competition, whereby the gut microbiome, they try to prevent the entry or the survival of incoming pathogens through nutritional competition. They make sure they finish the nutrient or they exhaust it so that the incoming pathogens does not survive. There is also the niche inclusion. Like I say, niches are small habitats. So once they occupy those physical niches, they deplete essential nutrients. And also, some of them produce inhibitory metabolite that stops the incoming pathogen, or possibly antimicrobial resistant, antimicrobial carrying of pathogens from surviving. There is also the contact-dependent inhibition that enables bacteria to outcompete rivals through direct cell-to-cell interaction. Next, further mechanisms. Next slide, please. Includes inhibitory metabolites. The gut microbiota, for example, produces some inhibitory metabolites like the short-chain fatty acid and bile acids that help in inhibiting pathogen growth. There's this natural mucosal barrier that protect habitat like the gut and the vaginal tract from pathogens. We have oxygen-limited conditions. Next slide. Next slide, please. Immune modulation and also antimicrobial peptides. The antimicrobial peptides actually, they exhibit bactericidal and anti-inflammatory as well as anti-endotoxin properties that serve as first line of defense against microbial threats. And these are a promising new class of antimicrobials that could address the antibiotic resistance crisis, which poses a major threat to human health. I have this table, next slide. I have this table that actually listed some of the antimicrobial peptides, where they are the microorganisms they are derived from, their mechanisms of action. Just go to the next slide, please. Some of them include niacin, bacteriosin, etc., etc. So what roles does the gut microbiota play in different types of MDR infections? Evidence have shown that the main role of gut microbiota in modulating the spread and progression of antimicrobial resistance, the gut microbiota microbial ecosystem can either facilitate or impede the colonization of drug-resistant pathogens. Just go on to the next slide. So this slide shows some of the resistant bacteria. Mostly I use the escape bacteria, or called the escape bacteria, and the role they play in MDR infections, like the Enterococcus faecal. This organism, the gut microbiota resists it by secreting antibacterial substances that inhibit its growth. Our own popular Staph aureus, the gut microbiota counters it by restoring the mucosal barrier and suppressing inflammation, thereby limiting bacterial colonization. We also have the Klebsiella, and that has its own role to play in curbing the spread. The gut microbiome has its own role by releasing beneficial metabolites that helps in curbing the spread of Klebsiella pneumonia. Then we have Pseudomonas, Acinetobacter, and then the rest of them. Next slide, please. So what are the targeted intervention that the microbiome uses for resistant mitigation? They modulate microbial communities, they influence pathogen survival and susceptibility to treatment. So the microbiome plays a crucial role in doing this. In addition, they possess the potential of altering the composition and activity of the microbiota to reduce the colonization colonization and spread of drug-resistant pathogens to restore microbial balance and improve health. Next, please. So some further strategies that can be used by the microbiota. The first one is the use of probiotics. So probiotics they are bacteria, but they are what we call beneficial or good bacteria that can be taken in known doses to restore the health of the microbiome. So restoring the health of microbiome, you know, I told you that antibiotics, when they get into the system, they first of all, they cause what we call dysbiosis. But using beneficial bacteria or beneficial microorganisms, it can be bacteria, it can be fungi. It can help to restore the health. And this will help to exclude resistant pathogens and reduce the reservoir of antibiotic-resistant genes in the gut. So, but when you use what we call the prebiotics, they are not organisms. They help the probiotics to actually be more efficient. And when the two of them combine, they form what we call the symbiotics. So prebiotics, they nourish the beneficial microbes, while symbiotics combine probiotics and prebiotics to produce what we call the synergistic effect. And the postbiotics, that is what provides antimicrobial and anti-inflammatory benefits. These strategies collectively suppress harmful bacteria, enhance immune responses, and maintain gut microbial balance. Next, please. Yeah, another intervention the microbiome can do is what we call the fecal microbiome biota transplantation. Fecal microbiota transplantation. This one is like a donation whereby there is a transfer of physics, healthy, we'll call it healthy physics, from a healthy donor that has been minimally processed. We send it to be given to a patient that has severe, maybe diarrhea or severe dysbiosis, that his gut lacks normal bacteria or normal microorganisms that will help it. So it is usually given. There's a place I gave a talk and told them that you can transfer physics into a sick person from a normal person, a healthy donor. Physics can be donated. People were asking me, how can somebody eat physics? Is it not excreta? How can? So that's why you can see that it can be formed into capsules rather, that will be taken and opens when it gets to the intestine, where it does what? It restores the microbial diversity and provides the balanced functional microbial community that is needed for a healthy gut. So this restoration enhances colonization resistance to multiple drug-resistant pathogens through mechanisms like competitive inhibition, bacteriosin production, and immune modulation. Next, please. Next. We have what we call the vaginal microbial transplantation. So there has been growing interest in vaginal microbial transplantation. And these workers reported the use of vaginal microbiota transplantation from healthy donors to an as an alternative to the treatment of patients with bacterial vaginosis. So there's a research we did that whereby we examined some women, vaginal swab from some women, and observed that some of them had bacterial vaginosis. Five out of the patients with positive PV cases were used to do what we call the yogurt experiment. We prepared yogurts. It was part of one of my PhD's work and a little aspect of her work. So she prepared the yogurts where we added live probiotic organisms, lactoplantin, Bacillus pentosus, KCA1. That's the name of the bacteria. It's a lactobacillus species. We prepared it and actually gave it to these women, 200 ml, for a period every daily for 2 weeks. At the end of the 2 weeks, 3 days after, the vaginal swabs were collected again and re-examined. 2 of them showed sign of clearing of what we call clue cells. Clue cells are evidence of presence of a bacterial vaginosis. 2 of them showed the evidence of clearance of clue cells, and the vaginal swab smear was showing more lactobacilli species, showing that normalcy has been restored to those bacterial vaginosis patients. Next slide, please. You can also use bacteriophage therapy as a microbiome intervention in taking care of multi-drug resistance pathogens. What are bacteriophages? These are viruses. They are microorganisms. They are viruses, but their job is to infect and lyse bacterial cells, thereby offering a targeted alternative to traditional antibiotics. So these pathogens, they have the potential to do what? To selectively lyse pathogenic bacteria. So when they see pathogenic bacteria, they lyse them. And by so doing, they reduce as much as possible the resistant carrying bacteria. So this is a key advantage over broad spectrum antibiotics. Next, please. So still on the strategies to combat resistance. Targeted modulation of the microbiome, for instance, the fecal microbiota transplantation has shown promise in decolonizing multiple drug resistance organisms and persistently reducing antimicrobial resistant genes in several clinical studies. Microbiome-based therapy may enhance immune responses and in turn reduce the need for antimicrobial agents, which helps slow the spread of resistance. Next. Next, please. So preventing, wow, go back, please. Preventing antibiotics-induced dysbiosis using dietary modification and microbial intervention supports a resident microbiome able to resist pathogen colonization. So when you use this modification, dietary modification, the microbiome become more stable, and with that they are able to resist pathogen colonization. Also, increasing microbiome diversity through fermented foods, that is probiotic foods, prebiotics, and selective microbial supplementation can naturally impede pathogen growth and reduce dependence on antibiotics. So the applications of microbiome to antimicrobial stewardship, how does it apply? So probiotics and live biotherapeutic strategies introduce or enhance taxa that suppress pathogens, reducing need for broad spectrum antimicrobial agents. Microbiome guided dosing and stewardship tailor antibiotic choices and durations based on patients' microbiome profile to minimize collateral dysbiosis. So you see that the main aim is to actually reduce dysbiosis as much as possible. Then thirdly, you can use biomarker-guided intervention. So you use microbiome and the host gene expression signatures. You watch out so that it will help to predict susceptibility and response to treatment. Enabling targeted therapy. Therapy needs to be targeted, not just broadly wiping away everything that is in the gut. Number 4, environmental microbiome management. There's need to monitor the environment and mitigate antimicrobial resistant genes that are reserved in some parts of hospitals and agricultural settings to reduce dissemination. And then next, please. So what are the innovative things that can, and the future looking ahead going forward? You can manipulate, you've seen that manipulating microbiota composition using synthetic communities, phage therapy, or nutrient shifts are beginning to emerge as techniques to fight resistance. So these options can be explored. This can be achieved by suppressing the antimicrobial resistant gene carrying bacteria or restoring a healthy competitive balance. So there's also the possibility of using vaccine that protects against pathogen or target resistant conferring elements in bacteria. This could further reduce the burden of resistance and lessen antimicrobial impacts on microbiota. Next, please. Next, please. Next. Conclusion, we are concluding now. The widespread overuse of antibiotics disrupts the microbiome's balance, causing dysbiosis and promoting the spread of resistance genes, as well as increasing infection risks. The microbiome have been implicated as an alternative option in handling MDR, as the microbiota have different strategies they use in combating MDR infections. And finally, next please. A range of therapeutic approaches, including probiotics, FMT, VMT, and bacteriophage therapy, show promise in modulating the microbiota to enhance host resistance. These interventions aim to restore microbiome balance, strengthen protective functions, and mitigate AMR effects. So harnessing the potentials of the microbiota to enhance colonization resistance offers a promising avenue to combat MDR infections and mitigate the global threats of antimicrobial resistance. Thank you. Thank you for listening.
For Professor Agbakoba, you all can agree with me that she did justice to the topic as an expert in antimicrobial resistance and multi-drug resistance. Yes, we know that the problem of multi-drug resistant infections is escalating by the day. So she carefully, gently, step by step, took us through the strategies, the intervention strategies and ways to combat multi-drug resistance. Among many, she mentioned probiotics, prebiotics, symbiotics, postbiotics, bacteriophage therapy, and microbiome transplants, among many. Not just mentioned, she took us through them very well. Thank you so much, Prof. We appreciate you. We have questions. We have questions for Prof. And I'll read them. An anonymous participant said, "Dear Prof, do we have currently any probiotic product to manage colorectal cancer?" That's one question. Another question says, "Can probiotics alone be used for the treatment of multi-drug resistant infections?" Another person said, "Is there any commercially available human phage therapy today?" You can answer, Prof. Should I repeat them one after the other?
Please do.
Can I?
Please repeat them. Yes,
okay, maybe I'll take it one. Maybe we answer one after the other. The first person says, "Dear Prof, do we have currently any probiotic product to manage colorectal cancer?" Thank you.
Okay. Yeah. Research is going on from what I read and from what I know that some probiotics can take care of cancer. But I don't know whether they are currently available or they are still at the research level. That is what I cannot say, but I happen to know that such is on. But you know, cancer, well, they don't really have cure as to final cure. It's just managing and managing and managing. Management is what they use. But research is going on.
Thank you so much, Prof, for that candid answer. The next question says,
okay,
can probiotics alone be used for the treatment of multi-drug resistant infections? Probiotics alone.
Well, in some cases, something like in colitis, whereby you can use these fecal transplants. You can use the fecal transplantation to actually normalize the microbiome.
Okay. Thank you, Prof.
The organisms there. Hello?
I can hear you, Prof.
Okay. Yes, I was saying that although combining it with antibiotics helps, but where all the antibiotics are resistant, probiotics alone can help, especially in cases of diarrhea, whereby you use something like the FM, fecal microbial transplantation, to restore, because you know most of the organisms there maybe some of the organisms there may be probiotics, organisms in the stool, in the intestine, may be probiotics. So I believe that MDR alone, probiotics can take care in some cases, because when we used it in our vaginal experiment, that is the one we used yogurts to perform, it actually cleared the bacterial vaginosis infection that the women were having. Some of the women were having, not all of them. So I believe.
Thank you so much. Thank you so much, Prof. The last question is this. Is there any commercially available human phage therapy today?
I don't think so yet.
Is there any commercially available human phage therapy today?
At the research level, I don't think so. That's my answer.
Okay. Thank you so much. Another round of applause for Prof. Thank you, thank you, thank you, thank you. Now it's time to present the award for Prof. But please, before we go, we should remember that Prof advised antibiotic exposure should be minimized if it cannot be avoided. Thank you. To present the award is Dr. Mutiat Ibrahim. Presentation of award to Professor Agbakoba. Thank you.
Thank you. I'm accepting it so without seeing it.
Thank you, Prof. On
Thank
behalf
you very much.
of Lagos Faculty of Pharmacy, we would like to present this award to Professor Ineka Regina Agbakoba as a plenary speaker for the 4th Faculty of Pharmacy Virtual Scientific Conference. Thank you so much. Here is the presentation, which will be
accept it.
in a month or two. Thank you very much, Prof.
Thank you very much. Thank you.
Thank you.
God bless you. And thank you for. Thank you. And please pardon my network issues.
Thank you, Prof. We understand. It's Nigerian.
Yes. I'm telling you. Thank you for understanding. God bless you.
Round of applause for Prof. The next plenary speaker, we are still talking translational research. The next plenary speaker is Professor Mohammed Baraka. Distinguished Professor Mohammed Baraka will be speaking on digital health and pharmacy, transforming patient care with technology. And to read the citation of Professor Baraka is Dr. Ebele Onwuchuluba. Thank you. Kindly turn on your video while the citation is being read, Prof. Thank you.
Thank you very much. Good morning, esteemed participants. It is a great privilege to stand before you this morning to read the citation of Professor Mohammed Baraka. Dr. Mohammed Baraka is a distinguished professor and thought leader in medical, pharmacy practice, digital health innovation, and AI integration. With a strong background in healthcare technology and education, he's actively shaping the future of pharmacotherapy and pharmacy through research, teaching, and digital transformation. He has expertise in digital health and artificial intelligence and has received certification in applied digital health ethics and healthcare delivery value. He's a certified trainer in digital health in pharmacy education, was a speaker at the 7th GCCE Health Workforce Development Conference. He's an advocate for AI-driven clinical trials, digital therapeutics and automation, and an expert in informatics and AI-driven decision support systems. He's a pioneer in AI-enhanced medical and pharmacy education and clinical decision-making, a contributor to International Faculty Development Program in Digital Health, a leader in OX development and pharmacy curriculum innovation. He's a co-developer of the FIP Digital Health in Pharmacy courses, a member of the National UAE IPE Task Force for Interdisciplinary Healthcare Education. His current focus areas include AI integration in medical, pharmacy education and practice, digital therapeutics and patient engagement technologies, pharmacy automation and informatics for improved patient care, faculty and workforce development in digital health education. Professor Baraka remains dedicated to advancing medical and pharmacy practice through innovation, research, and digital transformation, ensuring that the next generation of healthcare providers is equipped for the evolving healthcare landscape. Dear participants, kindly join me in welcoming Professor Baraka to the podium.
Hello, everyone. Can you hear me clearly? Can you hear me?
Loud and clear.
I'm going to start my presentation now on my side.
Yes, please.
Can you see my slides, please?
Yes.
I'm now in presentation mode.
Line show, yes.
Well, great.
Awesome.
Hey, everyone. Good afternoon from the United Arab of Emirates, and good morning for you in Nigeria at the moment. This is Dr. Mohammed Baraka, Associate Professor of Clinical Pharmacy and former Dean of Pharmacy at Fatima College of Health Sciences in the United Arab Emirates. This is a great honor and privilege for me to be among this group of eminent professors and speakers from all over the world. Before I start, I would like to thank my colleague, Professor Ibili Onwacholoba, for extending the invitation for this conference. Since April last year, we have started this communication. I would like to extend my huge congratulations for the Dean of the College, Professor Sunday Edisigan, and my grateful thanks to Professor Margaret Sofidia and my colleague, Dr. Arinola Joda and Dr. Ebeli, who are also my colleagues in the academic pharmacy section at the International Pharmaceutical Federation. Today, we'll start talking about the digital health and pharmacy. Why digital health is very important nowadays for pharmacy education and for pharmacy practice, and how it is transforming patient care. And before I start, this is a brief about myself. I'm originally Egyptian, received my education in Al-Azhar University, Cairo in Egypt in 2003, and then moved to the Free University in Brussels in Belgium, received my PhD in clinical pharmacy in 2011, and then back to Egypt, 2 years as assistant professor of clinical pharmacy, then to Saudi Arabia as a founder and head of pharmacy department in College of Clinical Pharmacy, then to Al Ain University in 2020 as associate professor of clinical pharmacy, and now at Fatima College of Health Sciences. So today we are going to discuss about the changes happening in the healthcare space due to digitalization of health services and products. And we'll discuss about the core technological advances, what digital health is, and the subcategories under digital health, and why digital health is going to change completely the pharmacy practice. And this is a future that is already here. We'll also discuss the impact of digital health on the future of jobs and education. And hopefully, this will be a kind of eye-opening presentation for my colleagues and for students attending this conference to think about their future and to think about new directions for this future. Before I start, as a disclaimer, I may refer to some software programs, but this is solely for educational purposes. I'm not connected to any of them by any business relation, and I'm not promoting any of them. Today, we are living in a wireless connected world in which we are having very new tools and very new communication devices that we are using for example, for shopping, for games, for social media and communication. Even we're using them in online education, in telecommunication, and this is going to change the whole world. Healthcare is not an exception. It's going to be transformed completely as well because of this technological invasion of this field. In 2021, the International Pharmaceutical Federation has published the development goals of the Federation. And here we can see digital health clearly mentioned as one of these goals, in addition to many other important goals that are emerging, like pharmacy intelligence, like people-centered care, like competency development, working with others, continuing professional development, impact and outcomes. So if we talk about digital health, which is a major focus of our presentation today, and talk about what happened in the world after the pandemic in 2020, we can see here in a report published by one of the largest companies in clinical research, the IQVIA Institute. And here they started to collect and talk about the digital health tools that have been used heavily during the pandemic. Here we can see the smartphones using their cameras to capture skin lesions, for example, sending them to the doctors, having diagnosis from the rest of our homes, using telemedicine and virtual care in providing a kind of hybrid care. And by the way, hybrid care is the future. You cannot expect to go to the hospital or to go to the primary care center for every single complaint. There are so many things that can be dealt with virtually from the comfort of your home. And here in United Arab Emirates, last year in Dubai, only in Dubai, 375,000 virtual care visits have been accomplished here. And this is telling us how the future will be for the healthcare provision. Digital therapeutics, and we'll talk about digital therapeutics today as one of the major advances that are booming now. And the investment in this category of digital health is booming now. And it's being used in some countries, like I will talk now. Clinical trials is not centralized anymore. Clinical trials can be done from the comfort of the patient's home using virtual care and using sensors and wearables to collect data from patients. No need for a patient to travel 100K kilometers to go to or to participate in a clinical trial anymore. There are so many other digital health tools that I'm going to simplify for you now. So these are the major categories of the digital health. And digital health is the umbrella that includes all health tech that are transforming completely the healthcare. Look at the first one, which is telehealth, or we have nowadays telepharmacy as a special case of telehealth, virtual care, providing pharmaceutical care for patients from the comfort of their home, mobile applications, and what we call nowadays digital therapeutics, using software programs to manage, treat, and prevent certain diseases. Look at the robotics and automation. We know that there are smart pharmacies nowadays are run completely with a few robots, with one robot and a few pharmacists working together. Virtual reality that can be used, was used in the past for games, but now we use it in clinical training, in medical education, and we use it also in treating certain diseases, like I'll mention in a couple of minutes. Farmbots that started as chatbots in different websites. Nowadays, pharma companies are trying to use farmbots instead of medical representatives. Sensors, wearables and sensors have been one of the major digital health categories nowadays, with the advances of AI and sensors and all these things. So now with the Internet of Things and the Internet of Medical Things, you can use sensors to collect a huge amount of data about your patients that can be streamlined 24/7 to their healthcare providers. And then the voice assistants. We all have voice assistants on our smartphones and computers and laptops. And these voice assistants are not only here to talk to you about the weather or the news, they can be used also in medicine, like we will see in a couple of minutes. Artificial intelligence. Artificial intelligence is the major category that is taking all the traction nowadays in digital health, because it's a huge potential to improve and to completely transform healthcare provision. So how digital health works? If you start with the first subcategory, which is telehealth, we can see here a kind of remote patient communication using telecommunication tools like phones, like video calls, and providing virtual care for patients without the need to come to the hospital. Imagine a patient who just want to refill a prescription. Why they need to travel to the hospital, wait for 1 hour to have a new prescription to refill the ongoing medications. Now, telepharmacy is one special case that we can use to provide pharmaceutical care services. And there are so many pharmaceutical care services that can be provided using this important tool. You can do comprehensive medication management. The majority of our work is a kind of cognitive work, especially the clinical ones that have direct patient care, comprehensive medication management, medication selection and dispensing, refill of ongoing prescriptions, compounding verification for sterile and non-sterile preparations, patient assessment and evaluation, patient counseling, especially with the home delivery that tools that we have nowadays to send medications to patients at home, medication reconciliation, clinical consultation, outcomes assessment, and follow-up for our patients that we lose. We lose a lot of opportunities to follow up our patients after providing unique pharmaceutical care services to our patients, and then we lose them. And then we don't know what happened with the patient. And we also lose a great opportunity for experience building from the patient assessment and assessment of the outcomes. Second category is one very important category, which is digital therapeutics. And digital therapeutics are applications that you can prescribe for your patients. And it's happening nowadays. You might think this guy is coming to talk to us about something from another planet. No. Today in Germany, if you have some friends working as pharmacists in Germany, they sometimes receive prescriptions that contain conventional medications, in addition to some software applications to enhance the impact of medications or to help in adherence or to collect data about patient outcomes and to send this data to the physicians. And these digital therapeutics are going to be a kind of games that patients play and enjoy in addition to their medications. And sometimes they completely replace medications if they prove clinically effective in clinical trials. Yes, you heard it right, clinical trials. These tools and these digital therapeutics are different from the health apps that we have nowadays. Every one of us is having a mobile that is giving you access to more than 355,000 applications, software applications on the internet, only in health and wellness category. But digital therapeutics are different. Digital health therapeutics are software applications that have been thoroughly investigated in clinical trials, exactly like medications. So we do clinical trials to collect information about their safety, efficacy, and about whether they are really serving the purpose for which they are intended. And then they go to the regulatory affairs, to FDA, EMEA, to the DIGA program in Germany, and to other regulatory affairs in different countries to get regulatory approval to be prescribed, dispensed, and reimbursed by insurance, exactly like medications. This is no surprise, guys. This is a reality. And in Germany alone, there are around 55 or even more, 55 digital therapeutics that have been approved, prescribed, and dispensed, and also reimbursed by the insurance companies for patients. EndeavorX is one of these applications. It's a kind of video game that's used for children to improve their cognitive impairment, and it's FDA approved for treating attention deficit hyperactivity disorder. The third category is sensors. And we all heard about the sensors that we can use nowadays, like glucose continuous monitoring or continuous glucose monitoring, the CGMs sensors that we use to collect data about patients, about their blood glucose level 24/7. The smartwatches, and just yesterday or the day before, Apple announced that their smartwatch can measure the blood pressure, and it's regulatory approved now to measuring the blood pressure and to give you alerts about changes in your blood pressure. Other smartwatches that can measure and depict your ECG and give you information about how your heart is functioning. There are also bracelets that can be used in conjunction with some software applications to measure your blood pressure. And these wearables and sensors are also going to change how we perceive our patient's health, because it can streamline information 24/7. And all of us will be overwhelmed with the output and the data streamed for us from our patients every day. Robotics and automation. For example, here in United Arab Emirates, we have many smart pharmacies that are run by one robot, like you see here on the image, in addition to some few pharmacists. And there are so many smart pharmacies available nowadays in different countries, not only in the United Arab Emirates. Virtual reality that, as I told you, was used in entertainment, now is used in medical training and in virtual meetings. Not only this, but there are companies who received FDA approval for using virtual reality to treat certain diseases, like using EaseVRX, virtual reality goggle, to treat chronic lower back pain, as you saw here. Voice assistants. There are so many voice assistants that are, by the way, used in many companies, like you see here, like Google Assistant, Apple Siri. And nowadays, this is quite old statistics, but nowadays we know that so many companies are using voice assistants, and they can hear what we are talking about, and they can customize ads, and you can use it also for healthcare provision to improve patients' adherence by sending reminders and talking to patients about whether they have received their medications or not. Pharmacy chatbots, or what we call pharmabots. And pharmabots are trained nowadays to talk to patients, to give information about the medications available in the pharmacy. You can use it for advertisements, for some health and wellness programs you are developing in your pharmacy. You can also use it for answering some easy questions or frequently asked questions for your patients. Some of them are AI supported, so they can be smart and intelligent in answering different patients' questions and redirect them to human agents if necessary. So this is a chatbot that you can, these are examples for chatbots that can have conversation with patients, and we saw a lot of them during the pandemic and after the pandemic. Artificial intelligence, last but not least. Artificial intelligence refers to the ability of machines to simulate your intelligence, to think like you, to take decisions. And nowadays, we have the AI agents that can provide, even they can perform some tasks without going back to you. So you can program them to do some tasks and to work together in harmony. So I will not be surprised in the near future to see some of these AI agents working with us in academia, as I will show you here some examples of AI agents that are already working in medical and in pharmacy practice. So there are so many companies that are using AI, like Facebook, Gmail, Google. We all know that we can talk together now about restaurants, or we can discuss now that we are hungry after this conference and want to go to the lunch. Unfortunately, I will not be able to join you in lunch. We can do this virtually. But if we discuss this now, when you look at your Facebook account after a few minutes, you'll see a lot of suggestions. And this is coming from the AI that is hearing our conversation and customizing ads and advertisements regarding what we need to do and directing us even to take certain decisions. This can be used also in medications and AI-assisted drug discovery and the drug delivery or drug development is one of the very important applications of AI in pharma nowadays. We heard about some companies that are using or experimenting using AI to develop certain medications for certain receptors, for example, by simulating how AI can give you information about certain medications and how it is going to fit. So instead of doing a whole experiment that costs you 1 year of research in the lab and a huge amount of money to just to check one compound and check whether it is. And I know our colleagues in chemistry, medicinal chemistry, they can synthesize huge amounts of medications or chemicals, but they don't know, or they need a long time to check them biologically to know whether they will be fit or not. They can use some software applications to help them in fitting, but nowadays AI can simulate this and can give them enough information about the best leads that can be used instead of wasting time and money and research labs and making us busy for compounds that cannot be successful in the future. Other companies, they can map this against multiple genes to check for specific medications that can treat some genetic disorders. Here in the United Arab Emirates, we had the first AI minister dedicated only to AI in 2017. And France was the second in the world to do this, but 7 years later. I think this is a very important step for countries to think about because of the huge amount of investment that the whole world is investing in AI and its applications in medicine and in other things. So after giving this overview about digital health and its applications in healthcare, let's think about the future of pharmacy practice and how it is gonna look like in the near future. And here I'm not talking about the future, I'm talking about the past that has already happened. For example, here in the United States, CVS, one of the biggest companies or pharmacy chains, they decided to close 900 stores. And here they're closing physical stores only, but they are smart enough to move this business to the online and e-marketing and e-health provision to their patients. So think about this. If you are, for example, as a pharmacist or a pharmacy student attending our presentation today and thinking about your future, you might be thinking about migrating to the United States or Canada to work in one of these pharmacies. Think about preparing yourself to that role, not to be a regular pharmacist. You need to be a digital pharmacist to leap into these new expanded roles for pharmacy. And here we are having a new diagram for the patient-centered care for pharmacists, and talking about collecting data, assessing patients' data, providing pharmaceutical care plans that are evidence-based, implementing this and monitoring and evaluating patients. And here we are talking about the implementation of AI in all these steps. I don't have time to discuss all this, but there are so many applications that we can discuss in the Q&A session if we have time. Think about this. This is an autonomous car that is sending medications to patients to home by home delivery without having anybody to accompany it. It's an autonomous car. And here we are sending medications. There's one of the hospitals here using it to send medications to patients in the smart city in Dubai. And the same hospital is piloting sending medication by drones to patients as home delivery too. Amazon has already started this business in the United States, and it will come to you, and it will come to us here, and it will go everywhere to do the same and expand the same business. They are using drones and they are using autonomous cars to deliver medications to their patients. So think about this future. I believe the future is for digital health, for e-pharmacies, for e-prescription, and for hybrid care. So if you want to get something from this presentation, I need you to think about your future as a digital and e-pharmacy and prepare yourself for these roles. You need to think about co-working with AI agents and AI tools. For example, what you see here on the screen is AI agents developed by one of the US companies to provide perform certain tasks for patients. And today you can recruit them to do some of your work. So here, for example, Mira can do remote patient monitoring. Erin can help in transition of care chicken. And these are not humans. These are AI agents that are very well trained to do these jobs. And I saw some of them are doing patient reconciliation. Some of them are doing patient consultation. Some of them are providing pharmacy teleconsultation. And you can see pharmacy category here. You may check it after the presentation. All these categories will help us to collect 360 information from our patients about precision medicine. So you can use AI to collect information and process it, and add to this the clinical genomics and the pharmacogenomics to include information about the patient to provide precision medicine. Not only precision medicine, but precision health. Look at this image. Here you can collect whatever information about your patient using a holistic approach to collect information about the environment, about the biomarkers, about the clinical measures, about the social determinants of health. Use the digital health tools, sensors and wearables and digital therapeutics, and utilize all this data to provide customized precision health that is really tailored for your patient in the future. Add to this 3D printing, and I know some companies and some pharmacies for the first time in the world, last few months, they printed medications for their patients in the community pharmacy. So for students attending with us today, you can expect your pharmacy in 10 years from now to have this 3D printer to develop such medications for your patients that are very appealing, containing only the exact amount of active ingredient for your patients, customized for the patient, age for the patient, weight for the patient, palatability and flavors they like, especially for children, the colors they like, for everything. And you can print these medications in the pharmacy like they did in Spain a few months ago. So while exploring all these opportunities, we should stop here and ask ourselves, especially as academicians, are we still doing the right thing, teaching pharmacotherapy and other clinical pharmacy and other pharmacy-related courses the old way? I believe not. I think we need to revolutionize our curriculum. We need to change what we are teaching, how we are teaching, how we are training our students, how they are going to utilize the huge amount of data coming in the future, and how they are going to make sense by introducing such things to the patient workflow. Think about this. This is a publication from Peterson Health Technology Institute. And here they are talking about the huge amount of data you can collect about diabetic patients using several tools that will be processed by some AI agent, some AI tools, and in addition to humans to stay in the loop as well. But this type of humans should be different from the old people, because in the future we will need digital pharmacists who can utilize such tools and can be co-working with these tools to process the data that we will receive from many different sources. And now we have AI tools that are multimodal, or AI models that are multimodal, and that can deal with the different sources, whether it is images, text, voice, or any type of data, and can process all this together to give you recommendations. Even during the recommendation phase, some digital health technologies can help in the clinical decision support and in the decision-making process to achieve your goals and follow up, also using some digital tools. And for that reason, the digital health management solutions, in my opinion, should be part of the curricula in diabetes management. For example, I'm not talking only about diabetes, I'm giving this as an example, but there are so many software applications that are used nowadays in different countries to treat different diseases. And I believe the future should be different in teaching and learning, given that we need to integrate these things in our curriculum. So what we need to do, this is a wake-up call for my colleague academicians and for everyone. We need to respond to this, and we need to change the way we treat. As of right now, the problem is that the higher education is not set up to teach or train or educate people for such work, unfortunately. And this is a problem for the majority of academic institutes worldwide, by the way, not only in certain countries, but all academicians are struggling with the new reality. We should teach this technology to be a good team player and be ready to be part of the team. We will be working in a set like this in the near future, working in the lab while a robot is doing some experiments with us in the same lab. And to do that, the FIB, or the International Pharmaceutical Federation, started to draft what's called a Global Competency Framework. And here, for the first time in 2020, they put what's called digital literacy as one of the competencies that the graduates should enjoy during or after graduation. And then in 2021, they drafted what's called FIB Digital Health in Pharmacy Education. And here I had the honor to participate with some specialists from the different parts of the world to develop Train the Trainers course of digital health for pharmacists and for practitioners worldwide. And we also discussed again the digital health tools and as part of the academic capacity assessment tools. Here in Saudi Arabia, for example, they put in their PharmD learning outcomes the one thing about the digital health and utilization of technologies to optimize medication use. I also participated in developing the national academic reference standards for pharmacy education in Libya. And again, after checking all important references, we had to include digital health literacy as part of the learning outcomes that should be available in the pharmacy education. The American Association of Colleges of Pharmacy, they also updated the curricular outcomes. Instead of using CAPE 2013, now they are using what's called 2022 Curriculum Outcomes and the Entrustable Professional Activities, COBA. And this COBA will be adopted and adapted by all of us, especially those who are having American accreditation in our curricula very soon. And here they talked again about digital health and using digital technologies to improve patient outcomes. In United Arab Emirates, we had it also as part of our competency framework, utilizing technology, AI, human economic and physical resources to optimize medication use. And in order to fulfill the requirements of these standards, the national and the international standards, we had to include in our curriculum what's called digital pharmacy and the informatics course, in addition to digital health and automation training or internship for our students before graduation, in addition to many other important courses like precision medicine and other electives that are futuristic, like innovation and entrepreneurship and other, among other things. So how can we start to be practical? You need first to identify the champions in your college. You need to check whether some of them are having self-study pathways or are enrolled in digital health programs, having enough knowledge and skills, or you can develop an internal training program in your institute and help your faculty to have digital health education initiatives in your institute to drive the digital health and the integration of AI and technology and digital health in all curricula. And there's a kind of maturation process that you can do here. You can start by providing adjunctive degrees or elective courses, not necessarily to be built directly in the curriculum unless you have enough expertise and enough infrastructure, or it can be later as a part of continuing education initiatives that you provide, or can be a kind of postgraduate study or training, or can be integrated in such programs. Some universities started to revisit Bloom's taxonomy and to change learning outcomes based on what AI capabilities can do and based on what humans can do, and keeping humans in the loop. There are distinctive human skills and AI capabilities that can be combined to improve our teaching and learning. So the future and the current jobs in digital health. In the future, when you students here and early career pharmacists go to the interviews, you will be expecting to see those guys. You will not see them like this, but they will be AI agents. And according to McKinsey and company, there will be AI agents, they will be constituting around 15% of workforce in the future. So if I attend with you 2 years from now or 3 years from now, another edition of this conference, I will not be surprised to hear the dean talking about the workforce in the college saying, well, Dr. Baraka, 3 years ago, when you talked about this, we had 15 or 20 faculty staff in the college. But this year, we have 25 faculty staff and 5 AI agents helping us in providing high quality education for our students. I will not be surprised to see this because I hear this in the conferences, and I showed you already some AI agents who are providing such services in pharmacy practice. We will have some of them in education very soon, and we are developing some of them now for education to automate, especially student support. This is a list of future jobs that can be expected to emerge in the future, like pharmacogenomic consultant, clinical decision support specialist, telepharmacy specialist, health informatics specialist, and for that reason, AI-assisted drug development scientist. For that reason, I need you guys, the students attending today, to explore these areas, because you will be required to participate and contribute as specialists in these things within few to maximum 10 years. The Saudi FDA 2 years back launched licensing initiative for digital health developers. So the world is in need for digital health developers. The world and the pharmacy practice in need for telepharmacy specialists. Here, as I told you, in Dubai, 375,000 telehealth consultations only in 1 year, in 2023. Imagine this is growing with the number of software applications and telehealth tools we are using every day. This is another application that is used for patients for virtual visits. This is another application called the DAWAC that is used for delivering medication to patients at home, then providing teleconsultation with pharmacists after delivering the medications here in United Arab Emirates. These are snapshots from some companies' websites and from LinkedIn for jobs that are already existing. I'm not talking from another planet. I'm not talking about the space medicine. I'm talking about, however, it's a specialty nowadays, space medicine. I'm talking about reality, which is we need telemedicine product specialists. We need robotics engagement managers. We need people who can work with such tools. We need digital transformation and robotic managers. Some of those will be from IT background, but some of them, if you look here about the job and the qualification requires, we need people from future healthcare and pharmaceutical industry who are innovators and the dream makers to join and work in such. We need people who are combining between this digital literacy and the healthcare background or pharmacy background. These are all screenshots from LinkedIn. There are so many jobs I see every day that you will not qualify for these jobs right away upon graduation unless you have to study in certain other fields like digital health, AI, and all these technology applications in healthcare. So the typical questions that I expect from some of my audience here today, are robots replacing us humans? No, robots are not replacing us, but the reality is robots will be replacing the repetitive tasks that many of us are doing now and will be competing with us on these repetitive and data-based jobs. And the right thing we can do is to utilize them as our minds and as our brains for the future, to use them as tools to improve our productivity. You shouldn't think about them as threateners or challenge or things that are coming to take your job, but they will take your job if you stay traditional pharmacist. And here, again, this is a wake-up call for all audience to prepare yourself for this future that is already here. So finally, transformation through technology is changing healthcare to more personalized medicine and health, and integrating digital health to education is actually not an option anymore. We need to do this and include this in our curricula. We started this already here right after the pandemic, and we can help other colleagues in other parts of the world to develop such programs and tools and to develop training even in such programs. Emerging career pathways are coming our ways now. Look at LinkedIn and search on digital health jobs, and you'll find so many. The future of healthcare is digital, is hybrid, and clinical. So finally, thanks so much everyone for attendance and attention. I'm really grateful to attend this prestigious event and to share with you what the future is, going to do to transform our jobs and our career. Here is my LinkedIn page if you wanna contact for future questions or programs or whatsoever. Thank you very much. Appreciate it.
A round of applause for Dr. Baraka, an associate professor. You all can agree with me that he did justice to the topic. He is well learned in this digital health matter. He took us through the integration of digital health with pharmacy practice to overcome limited access to care, medication errors, and poor treatment adherence. And he charged us, the pharmacists, to think of the future as a digital pharmacist and working with AI healthcare agents. Though there are challenges, the future of pharmacy lies in embracing technology not as a replacement, but as an enabler, supporting pharmacists in delivering safer, more efficient, and more patient-centered care. And he charged pharmacists in academia to adjust what they are teaching, how they are teaching, to incorporate the use of digital technologies. Another round of applause for Professor Baraka. I have a question here for you. We have a question for you. Two questions. The first question says, thank you for your presentation, very interesting, and it was a highlight in the recent concluded FIP. My question is this: if the Faculty of Pharmacy were to integrate digital features into their curriculum, would it be more effective to house this initiative under an existing department, or would it be more beneficial to establish it as a standalone department, such as a department of digital pharmacy? Or should there just be a feature of it in all the departments? That's the first question.
Well, great question. This is a $1 million question, and it indicates that our audience are now aware that digital health is very important for our future, and digital pharmacy and AI integration in the curriculum is not an option anymore. It's a kind of obligatory responsibility on academicians nowadays to adapt and adopt these tools in our curriculum. And the answer is, it depends. It depends on the infrastructure we have. It depends on the champions we have in the college. It depends on the human resources and capabilities we have. What I would suggest first is, number one is to identify the champions, the early adopters in the college who are willing to take this initiative and to implement such thing. And to be honest with you, all of us should be early adopters now, because AI and digital health is going to replace us also in academia if we don't come up to this challenge. AI agents and companies the problem is that, and this is for my academician colleagues, the problem is that the big companies like Google, like Oracle, like Microsoft, they are having their own academies nowadays. And they started to provide training programs for job seekers so that they can provide you a kind of internship for 6 months, for example, focused on the skills they need, and then they give you a job right away. If they expand in this direction, we will be in trouble in academia. I believe the future generations will go to these academies right away because they provide them the best type of education that they need for the best type of jobs. And they will not go to the universities anymore. This can happen within 10 years from now if you don't move fastly. So the first thing is to identify the champions, to support them to attend conferences, to learn courses. I can provide help and support in this. There are so many free courses and there are so many paid courses, and it depends on how fast you want to implement this. The second thing is to think about transforming the curriculum by including AI tools in all possible areas that we can use in. So here I'm not talking only about clinical sciences, but I'm also talking about basic sciences in which AI tools can be used nowadays for so many applications. So using AI tools in, as we saw, for example, here, we saw the AI agents that can be trained and can be recruited to provide medication reconciliation, for example. So in my lecture about medication reconciliation, I should talk about these tools and how they've been used and how my students in the future who will provide medication reconciliation, how are they going to provide this service besides these tools, or if this is completely will be done by these AI agents, then I should be doing something else. So we should think about what will be completely replaced by technology, and we teach such technologies in addition to teaching our students and future generations how to work with this technology. What will be done by technology? So I leave it for technology, but I understand what's going on because in the future, you will be collecting data from your patients 24/7. And here again, you need to understand how to utilize this data to improve patient care. In the past, we were seeing the patient once per month or once every 2 months to ask about how was your blood pressure? And then the patient will say, well, okay, there were some ups and downs, but to what extent, I don't know. Today, with the complete glucose monitors, for example, you will think about trends. You will not think about snapshot of the patient health anymore. When the patient comes to you, he will come with the reading of today, or the reading of yesterday, or the average of last week. But the patient of today who is using the continuous glucose monitor will send you information through the software application on their phone that's connected to this sensor. They will send you 24/7 of their information. And here you will be able to see the trends, not just readings, because they are different. Having a blood glucose level today of 140 milligram per deciliter does not mean anything if I don't know that the day before it was 180 or it was 200 milligram per deciliter.
Awesome.
Or it was 90 milligram per deciliter. So
Thank you
this is the
so much.
future.
Thank you, thank you, thank you. We have 4 more questions. Not only was your lecture thought-provoking, your response is also thought-provoking. Thank you, sir. So the next question says, great presentation. Kindly comment, it says, kindly comment briefly on patient privacy and confidentiality in digital health.
Another great question. This is one of the biggest challenges that we are facing nowadays, especially with AI tools. Patient privacy is really very important. And if you have a software application that is brilliant and that you can use for collecting data from your patients, but it can be hacked easily, or it can breach the privacy of the patient, or it can give access for this data to other people easily, then here you are in trouble. First of all, you will not get regulatory approval for this because your application should be HIPAA compliant before you go to the FDA. So privacy is a challenge. Unless you prove for the regulatory affairs that the privacy, patient privacy, and security of the data are very important and you're taking care of this dilemma, then you will not get approval and your application will be just one of thousands or hundreds of thousands of applications nowadays. But you can trust the FDA approved digital therapeutics, the EMEA approved digital therapeutics, the DIGA approved digital therapeutics. This is why we have 355,000 of health and wellness applications. But in Germany, for example, we have only 60 applications approved. And those 60 applications approved for dispensing prescription and reimbursement, they have fulfilled this important criterion. Okay, so this is one of the challenges, and the ethical considerations should also be part of any curriculum that we study and that we teach about digital health, and we put it in the curriculum of such training programs, of course. Thank you for this.
Thank you so much, Associate Professor Baraka. Dear conferees, I hope that you are still networking, not only with your fellow participants, but also with the speakers. Our time is fast spent. We have some more questions in the queue, question and answer segment. Kindly type the answer for them in the segment. Thank you, Prof. We appreciate you. Thank you.
Thank you very much. It was a pleasure being with you today.
It's now time to present the award to Dr. Baraka. And to present this award is pharmacist Alex Akinola. Thank you.
Thank you once again, Professor Baraka, for this exhaustive presentation today. So, on behalf of the University of Lagos Faculty of Pharmacy, we present this award to Professor Mohammed Barakat as a plenary speaker in this 4th Faculty of Pharmacy Virtual Scientific Conference. Thank you very much, sir. And we promise that this plaque will get to you as soon as possible. Thank you so much.
Thank you so much, Pharmacist Akinola.
Thanks a lot, appreciate it. Thank you.
Response. Okay. Bye. It's now time for the oral presentations. And to introduce us to this segment, we call on Professor Abimbola Chuemimo. Thank you.
Good afternoon, distinguished ladies and gentlemen. Thank you for staying on. Today we have 5 presenters in the orals presentation segment. So what we did was to ask an assessment of the presentations, and for each segment from pharmacognosy, clinical pharmacy, pharmaceutical chemistry, pharmaceutical microbiology, and pharmaceutics, they were chosen, one abstract was chosen, judged as best, and was chosen for oral presentation. So this morning we have 5 presentations. The presenters are all online. So because of our time constraints, we'll take 3 presentations first. You'll ask your questions, then we'll take the last 2 presentations. Then you can ask your questions. So I would just give us a brief run of the first 3 presentations, their titles, after which the presentation videos will be presented to us. So the first one is antibacterial activity of methanol root extract of combretum against multi-drug resistant methicillin resistant Staphylococcus aureus isolated from wound infections, and GABA S will be presenting that. Following that, we'll have phytochemical in silico and fertility enhancing evaluation of a polyherbal aphrodisiac root extract in Drosophila melanogaster. That will be presented by Akintola J.A., I apologize. After that, we have comparative in silico evaluation of compounds of plant origin as potential anti-cancer agents via molecular docking and ADMET profiling. And that will be presented by Kutu S. We will play these three videos for you now. Please listen attentively, after which we will take the questions for the three presenters and they will be available to answer your questions. Then we'll take the last two presentations and take questions and finish the oral section. Thank you very much. We'll start with the antibacterial activity of methanol root extract of Combretum micrantum methicillin resistant Staphylococcus aureus isolated from wound infections. Thank you. Can Gaba, Akintola, and Kutu please indicate with their digital hands raised that they are present and ready for questions, as well as Adedikwe ME and Additional JE. Please indicate on the platform with your digital hand raised that you are present and ready for questions when they are presented. Please bear with us. The videos will soon be up. Thank you. Let me also use that opportunity to give you the title of the last two, so you also begin to prep. Safe CAPS, a community-engaged M-Health intervention to improve quality of life in Black prostate cancer survivors. That will be given by Adedikwe ME. Then we have development of Terminalia avicenioides extract hydrogel formulation for the treatment of uropathogenic Escherichia coli in catheter-induced urinary infections. Please put your questions for us in the chat box.
Video is muted.
Ladies and gentlemen, we'll be starting with the phytochemical in silico fertility enhancing evaluation of polyherbal aphrodisiac root extract in Drosophila melanogaster. Thank you.
Hello, esteemed audience. I'm Jamil Akinkwelu Akintola, speaking on behalf of my co-authors to a paper titled Phytochemical in Silico and Fertility Enhancing Evaluation of a Polyherbal Aphrodisiac Root Extract in Drosophila melanogaster. Background: declining male reproductive health characterized by reduced sperm quality and increased infertility rates affecting approximately 50% of cases globally has become a concern. Scientific focus has been mostly on female fertility, leaving gaps in phytomedicine for male reproductive health. This study investigated the fertility-enhancing potential of an aqueous ethanol root extract from a polyherbal aphrodisiac formulation comprising 5 plants, namely Nestis ferruginea, Dioscorophyllum cumensi, Trichlicia subcordata, Griffonia simplicifolia, and Bosca angolensis, using phytochemical, computational, and experimental approaches. Methods: root samples were authenticated at the University of Ibadan Herbarium and extracted using cold maceration with 65% water and 35% ethanol. Phytochemical screening, GCMS profiling, molecular docking with alpha adrenergic receptors were carried out. So, antioxidant capacity was evaluated via the DPPH assay. Cytotoxicity screening was done using the brine shrimp lethality test. Fertility studies was also tested using the Drosophila melanogaster model. Results: the extraction yielded 7.60% crude extract. The phytochemical screening revealed that the extract contains flavonoids, terpenoids, phenols, saponins, tannins, flavotannins, steroids, and reducing sugars. 11 compounds were identified in the GCMS analysis, with methyl benzoate being the most abundant at 65.10%. The antioxidant activity gave a dose-dependent DPPH scavenging ability at 50% inhibitory concentration of 180.32 microgram per ml. The in silico docking showed that methyl benzoate had a favorable binding ability at minus 6.256 kilocal per mole. The brine shrimp lethality assay showed that the polyherbal concoction is non-toxic with 50% lethal concentration of 393.1 parts per million. The survival and fertility study with Drosophila melanogaster showed the concoction reduced mating latency, increased offspring emergence, but decreased adult survival. Conclusion: the polyherbal formulation shows fertility enhancing potential via antioxidant mechanisms and receptor modulation. However, reduced survival rates highlights the need for thorough toxicological evaluation before therapeutic use. Here are a few of the references that were consulted. Thank you for listening.
Thank you very much. We'll now play 006. That is the antibacterial activity of methanol root extract of Combetrum micranthum.
Good day to you all. My name is Samira Garba from Ahmadu Bello University Zaria, and I'm here to present to you a research titled Antibacterial Activity of Methanol Root Extract of Combetrum micranthum against multi-drug resistance MRSA isolated from wound infections. MRSA in wound infections are problematic due to their ability to resist multiple antibiotics, making treatment options complicated. Therefore, there is the need for other therapeutic options to enhance patients' outcome and improve the quality of life. Combretum micrantum root is used to treat some diseases among Zaria populace. The brief description of this plant, the English calls it long life, the Hausa Giza, Yoruba Okan, and the Igbos Enza Otegu. It comes from the family Combatraceae. It's a shrub that has leaves, roots, barks, and twigs. It's widely distributed throughout West and Central of Africa, with Nigeria inclusive, and it has been found in the western part of India. It has been used traditionally in the management of liver, kidney disorders, fever, diabetes, and general wellness. The aim of the research was to determine the antibacterial activity of this plant against multi-drug resistance MRSA isolated from wound infections in patients attending Ahmadu Bello University Medical Center. Ethical approval was obtained from the Ethical Committee on Use of Human Subject for Research in Ahmadu Bello University with the approval number, and also Committee on Animal Use and Care for the acute toxicity analysis. The selection of antibiotics, at least one agent, was selected from 11 different antimicrobial categories that are used in the management of staph aureus. The research design: 100 non-duplicate suspected coagulase positive submitted to the medical laboratory department of this hospital were collected and transported down to pharmaceutical microbiology Ahmadu Bello University Zaria for further analysis. Identification of isolates were carried out using microbiological standard. Antibacterial susceptibility testing was carried out using modified Kibibua-Diggs diffusion method, and interpretation of results were based on CLSI standard. Now, to the MRSA isolates that are multi-drug resistance were subjected to molecular analysis. DNA extraction was carried out using DNA extraction kit and further subjected to polymerase chain reaction for detection of genes. On the other hand, the plant was abstained from Zaria, and authentication of the plant was carried out at the Herbarium Section, Department of Botany, Faculty of Life Sciences, Ahmadu Bello University Zaria. The plant was subjected to a cold maceration extraction using methanol. Antibacterial activity was carried out using agar-well diffusion, broth and agar dilution, and further studies were carried out preliminary phytochemical screening, and also identify the compounds using GCMS analysis, and also the safety profile using acute toxicity OECD guidelines. The results were analyzed and recorded as averages and percentages in tables, figures, and plates. Results and discussion. From the results, it was observed that 21% of the isolates were staph aureus. However, other coagulase positive were observed in this study, and the isolates subjected to antibiotic susceptibility testing reveals the antibiotic resistance mostly to sulfoxetine, amoxicillin, and doxycycline. Now, phenotypic evaluation of MRSA using sulfoxetine antibiotic reveals that all the isolates were resistant to sulfoxetine, making them MRSA positive. And however, 73% of the isolates were resistant to at least one agent in 3 or more different antimicrobial categories. But the genes detection reveals that 100% of the isolates were amplified with 16S RNA, 88% amplified with the NOC gene, and 50% amplified with the MECA gene. And the plates 1, 2, and 3 shows the electrophoretic gel of 16S RNA, the NOC gene, amplified from the staph aureus isolates. Urinary screening reveals the presence of primary and secondary metabolites, which could be responsible for the medicinal property of the plant. And the in vitro antibacterial activity of the plant reveals activity at different concentrations, from concentration 12.5 milligrams per ml, and activity was observed to increase with increase in concentration. GCMS analysis reveals the presence of 9 chemical compounds, and then the identified compounds were mostly fatty aldehydes, fatty acid, alkaloids, polyphenols, and epoxides. And these compounds were reported to have a strong antimicrobial activity, even against other organisms. The acute toxicity testing reveals, even at the limit test dose of 5,000 milligrams per kg, There was no mortality, and there was no any significant alteration in their behavior, signifying that the plants are safe. In conclusion, the methanolic root extract of this plant has significant activity against multi-drug resistance MRSA isolated from wound infection at the MIC of 12.5 mg per ml, and it's recommended that the chemical compound reported to be present needs to be isolated and tested against other pathogenic bacteria causing wound infection. Synergistic activity with other antimicrobial agents needs to be investigated. Here are some of my references, and thank you for the audience.
Thank you very much. We'll play the last video for this segment, which is the comparative in silico evaluation of compounds of plant origin as potential anti-cancer agents via molecular docking and ADMET profiling.
Good day, everyone. My name is Kutusira Moturayo, and I'll be presenting this work on behalf of my co-authors, Jolayemi Olaoluwa, Nkocha Andrew, Owolabi Roda, Blessing Titilayo, and Alaribe Stephanie from the Department of Pharmaceutical Chemistry, University of Lagos. Our study is titled Comparative in Silico Evaluation of Compounds of Plant Origin as Potential Anti-Cancer Agents via Molecular Docking and ADMET Profiling. Background. Cancer remains one of the biggest global health challenges. In 2022, the World Health Organization reported about 20 million new cases and nearly 10 million deaths globally. Although chemotherapy has been central in cancer treatments, it faces major challenges such as drug resistance and toxic side effects that reduces patient quality of life. This has sparked interest in plant-derived compounds or phytochemicals as safer alternatives. Many of these natural compounds already show effective and selective anti-cancer activity, making them exciting candidates for drug discovery. Methodology. We investigated 7 compounds, including alkaloids, anthraquinone glycosides, which are secondary metabolites of medicinal plants. These compounds include pseudocarpine from Carica papaya, niacin from Moringa oleifera, rain, emodin and aloemodin from Cassia senna, argmalicin and alstonine from Rauwolfia vomitora. These compounds were tested in silico against 6 key cancer-related protein targets, which are nuclear factor kappa B, NFKB, STAT3, signal transducer and activator of transcription theory, PI3K, phosphoinositide 3 kinase, EGFR, epidermal growth factor receptor, HDAC, histone deacetylase, and BCL2, B-cell lymphoma 2. We use Schrödinger glide for molecular docking to predict binding affinities to the protein targets and Swiss ADME and protoxetine for ADMET profiling. To benchmark our results, we compared with standard reference drugs such as elotinib for EGFR, TTI-101 for STAT3, vorinostat for HDAC, idelalisib for PI3K, bortezomib for NFKB, and venetoclax for BCL2. Results. Our docking results were encouraging. Rain, emodin, and aloemodin showed very strong binding to PI3K, with docking scores around -10, which was even stronger than the reference drug idelalisib, which has a docking score of -7.158. The more the negativity, the better the binding affinity to the cancer protein targets. Niacin performed consistently well across multiple targets, ranking among the best in 5 of the 6 proteins, and was particularly effective against BCL2. Now, to the ADMET analysis, which is the absorption, distribution, metabolism, excretion, and toxicity profile for the drugs, the compounds. The gastrointestinal absorption was favorable for most of the compounds, suggesting good oral bioavailability. The log-P and TPSA values were mostly within the optimal range, showing a good balance between lipophilicity and polarity. This means the drugs are likely to have good membrane permeability and sufficient solubility, which are important for oral drugs. Pseudocarpine ranked highest overall in Swiss ADME, followed by argmalicin and niacin, indicating strong pharmacokinetic suitability. The anthraquinone glycosides, rain, emodin, and aloemodin also showed good drug likeness, meaning their properties are similar to successful oral drugs. Moving on to toxicity profile, Rain and Emodin stood out as the safest, with LD50 values of 5,000 mg per kg and no predicted hepatotoxicity, neurotoxicity, or immunotoxicity. However, one common observation among all the compounds was predicted nephrotoxicity, pointing to a potential kidney safety concern that will need careful monitoring in further studies. In summary, our findings highlight Rain, Emodin, and Alemodin as promising anti-cancer candidates with strong binding affinities, good pharmacokinetic properties, and acceptable safety profiles. The next step is to carry out in vitro and in vivo studies to confirm this effect and evaluate safety more closely, especially the renal findings. If validated, this compound could contribute to developing next-generation anti-cancer therapies that are both safer and more effective. Thank you so much for your time.
Thank you very much, our presenters. We don't seem to have any questions in the chat box. It means that every one of us understood the presentations clearly. But if you still want to ask questions, please put them in the chat box. So without much ado, I'll continue with the last two presentations, and I'll give you the topics of the last two presentations. Safe caps, a community-engaged M-Health intervention to improve quality of life in Black prostate cancer survivors, and that one will be presented by Adedipe ME, followed closely by development of Terminalia avicennoides extract hydrogel formulation for the treatment of uropathogenic Escherichia coli in catheter-induced urinary infections. We have additional JE making that presentation. One more time, please, if you have questions, please put them in the Q&A box. Thank you very much.
Hello, this is Dr. Adedipe, and today I will be walking you through a presentation titled Safe Caps, Improving Quality of Life for Black Prostate Cancer Survivors. Prostate cancer disproportionately affects Black men with an approximate 60% higher incidence level, and survivorship is often marked by fatigue, pain, and stigma. Right. And in the US in particular, men from African descent and even Caribbean descent, they face unique challenges in prostate cancer, including higher rates of diagnosis at advanced stages and also increased mortality. And this has been attributed to factors such as genetic susceptibility, limited healthcare access, socioeconomic status, and detection. And disparities in these survivorship outcomes have really persisted for so long. Now, mobile health is promising. We know that there's a higher smartphone ownership among Blacks, and that can also reduce barriers and may also address stigma by offering private engagement. And the mobile penetration rate in the Black community is over 80%, meaning 83% of Blacks own a smartphone in the US. Right, and perception of stigmas that comes from threats to masculinity and also because of the involvement of sexual organs in the disease. This could be mitigated since interactions are not face-to-face if interventions are loaded on a mobile phone. So a platform to empower Black men to be active participants in their care as they journey through the cancer control continuum. So that's just why this was really needed. Now, study objectives is really to improve quality of life and also survivorship outcomes through education, symptom tracking, and behavioral support. And how did we plan to do this? Well, it's been a study that has been going on for the past 5 years in different iterations. We started with a scoping review, which was published, and the citations are embedded on the slides if you want to read more about how we did it, basically how we built the app. And this was followed by qualitative interviews with Black men who met those criteria, inclusion criteria, being Black, being diagnosed with prostate cancer. And we also developed a small cohort to test the appropriateness and also to lay down the wireframe for the app. And the results I'll be sharing is from the pilot test of the study, which was conducted about 2 years ago. It was a 12-week daily smartphone assessment, and the outcome measure was quality of life. Here's just a rough overview of what the app really looks like. And the scoping review, which we did, identify the gaps in existing survivorship apps. The qualitative interviews was willing to put in some of the relevant domains that would really support Black men with prostate cancer. So this was a co-creative process. And yeah, so here's just a quick snapshot of what the app looks like and the domains it measures. In the pilot study, we had 15 men who were on average aged 68 years, which is a little bit lower compared to the major demographics that are diagnosed with prostate cancer. So Black men tend to be diagnosed at a younger age. The engagement level with the app was high. We had about 89% daily smartphone completion. And the outcome measure, which was done through FACT-P, which is a functional assessment for cancer therapy, was the prostate cancer measure, showed a significant improvement in quality of life at the end of 12 weeks, which was also statistically significant. Now, the feedback emphasized both short-term and long-term goals. They wanted to see more support at the onboarding. They would have loved to see more customization, being able to chat with their other friends who are using the app, and also being flexible with some features on the app. So these are things we plan to work on the long term with future iterations of the app. In conclusion, from the pilot study, we conclude that the SafeCaps app is feasible and also culturally responsive. It supports quality of life improvements for diverse Black survivors. Now, given that this is a pharmacy-related conference, the roles for pharmacists or providers cannot be overemphasized. It can help in onboarding, medication support, and cultural responsive care. Future directions will refine the app, test long-term impact, and also expand survivorship integration. Thank you.
Thank you very much. We'll take the last presentation now. Please remember to put your questions in the QA section. Thank you.
Good day, everyone. My name is Joy Adeshina, and I will be presenting our study titled Development of Terminalia avicinoides extract hydrogel formulation for the treatment of uropathogenic Escherichia coli in catheter-induced urinary tract infections. Now, the background to the study. The rising failure of conventional antibiotics to treat drug-resistant strains of uropathogenic Escherichia coli, which is a major pathogen in catheter-associated urinary tract infections, is a public health challenge, leading to recurrent infections, contributing to the economic burden and poor quality of life. Now, Terminalia avicinoides plant extract possesses antimicrobial activity against various bacteria. Hydrogels that are developed from biomaterials have been found to have satisfactory bioadhesive, biocompatible, and biodegradable properties, and so they serve as drug carriers offering sustained and prolonged drug action. Now, the aim of this study, therefore, is to formulate a hydrogel containing Terminalia avicinoides extract and evaluate its antimicrobial activity against uropathogenic Escherichia coli. Now, the method. Terminalia avicinoides plant extract was obtained by maceration of the stem bark of the plant in 96% ethanol. Now, this extract was used to formulate the hydrogel. 5 formulations of the hydrogel were produced by altering the composition of the hydroxypropyl methylcellulose polymer as well as the extract. Now, the formulations so produced were characterized for their organoleptic properties, their pH, their viscosity, their spreadability, their extrudability, their swelling index, as well as their antimicrobial activity using the agar diffusion technique. Now, here are the results. In Table 1, we see the organoleptic properties of the hydrogel formulations. And from the table, we find that formulation 1 to 4 have colors that range from light gray to dark gray. Now, this could be attributed to the change in composition of the excipients that were used, while formulation 5 is white in color as it doesn't contain the extract. Now, all 5 formulations were smooth in texture. For figure 1, the evaluation of spreadability showed that formulation 1 had the best in terms of spreadability. In figure 2, we see the pH for the 5 formulations, and from the graph we see that formulation 1 all fall within the normal physiological pH of the skin. However, formulation 5 has a slightly acidic pH. Now, this could be a cause of concern for prolonged usage due to its ability to cause irritation. In figure 3, we assess the extrudibility of the 5 formulations. And from the chart, we can deduce that formulation 1 and 2 have good extrudibility. However, formulation 2 gave the best result. For moisture content in figure 4, we see that formulation 2 had the highest value. Now, this could be indicative of its hydration potential. However, it could be a cause of concern for contamination if the formulation is not properly preserved. In figure 5, we evaluated the swelling index, which is indicative of the polymer hydration capacity as well as its ability to enable drug release. And we discovered that formulation 1 had the best swelling index and therefore would release its drug more quickly. In figure 6, we assess the viscosity of formulation 2 and formulation 5 at different share rates. And from the chart, we can deduce that both formulations have a pseudoplastic flow in that as the speed increased, the viscosity reduced. Now, this gives an indication that the viscosity is good enough for application and we enable the hydrogel to stay on the catheter when applied, and it will also be easy to insert because the viscosity would reduce during insertion and it will enable proper insertion. Here in table 2, we present the antimicrobial activity of the extract and the hydrogel against uropathogenic Escherichia coli. And from the table and the figure below, we can see that the extract at concentrations of 100 mg, 200 mg, and 400 mg per ml respectively shows good activity against the bacteria. For the hydrogel, formulation 2 shows good activity against the bacteria. However, formulation 5 shows no activity, which could indicate that the activity is due to the presence of the extract, since formulation 5 does not contain the extract. The minimum inhibitory concentration was also found to be 0.8 mg per ml. In conclusion, Terminalia avicenoids-based hydrogel exhibits promising antimicrobial activity against uropathogenic Escherichia coli and may serve as an alternative treatment for catheter-associated urinary tract infections. Now, further evaluation and optimization are needed to validate its clinical applicability. Here are my references, and thank you for listening.
Thank you very much, ladies and gentlemen. We're pressed for time, and we do not really have any question in the Q&A box. But if you still have questions for the presenters, please put it in the chat box. The presenters will respond to those questions. So I want to say a big thank you to all our oral presenters. Thank you for making this conference a beautiful one. Thank you.
Thank you so much, Professor Abimbola Shoemimo, for that oral presentation segment. Kindly put your questions in the chat box, like she said, and the answers will be given by the presenters. Now it's time for the parallel session. It's going to be very interactive, informative, and thought-provoking. Kindly join the different breakout sessions. And to introduce us to this breakout session, Dr. Oloma Obani Okoma will come and give us the ground rules.
Good afternoon, participants. Thank you for being here with us up to this moment.
Quickly, we'll go to our parallel sessions. The Zoom rooms are open. Please choose any Zoom room of your choice. So we have pharmaceutics, pharmaceutical microbiology, pharmaceutical chemistry. Then we have clinical pharmacy. So we have 5 breakout room sessions. Please, when you are done with the breakout room session, come back to the main session for the closing ceremony. And also there we will find out who the best poster presenters are. So please do not miss this session. Once again, when you are done with your breakout session, please come back to the main session. Thank you very much.
processing, range, process.
Welcome back from the breakout room. I want to find out if Dr. Adedipe is on this call. Thank you for staying with us up to this point. In the 4th Scientific Conference of the Faculty of Pharmacy, University of Lagos. Now it's time for company presentation before the closing ceremony. After the presentation, we are going to take the awards, the best poster awards for the different departments and units, followed by the award for the special people. To present to us today is Dr. Adedipe, presenting on Cotaries Solutions. You can share your screen, please. Cotaries Solutions. Okay, yeah, she's sharing.
Hello, can you hear me?
Dr. Adedipe.
Can you hear me?
We didn't hear you before now. Just heard your voice now.
Okay, well, hello everyone. Greetings from South Korea. Dr. Kano, are you there? Dr. Kano. Well, she's my co-presenter and she's supposed to be, I think she's on the line as well.
You want Dr. Kalu to speak? Dr. Kalu is muted.
We're both presenting, so yes, we're both presenting, yeah. If you can unmute her,
Take him
please.
co-host. Just hold on a
Thank
bit.
you.
Thank
All
you.
right, thanks.
What name did he use to log in? We can't see
Chisom
Kalu on
K.
the call.
She's Chisom K on the call.
Chisom K. Chisom K.
Yeah. She's in the chat as well, yeah.
Thank you. You have 10 minutes, please.
Yeah, we know, yeah.
Hello, can you hear me?
Yes.
Yeah, loud and clear.
Okay, awesome. I'm sorry, I couldn't unmute myself before I share my video. Good day, my name is Dr. Chisom Kanu, and I'm presenting on behalf of the company called Cotaries Solutions. So myself and my partner, Dr. Adedipe, are co-founders of this healthcare research consulting firm. Next slide, please. And so we are a healthcare research and consulting firm, like I mentioned, that committed to connecting diverse sectors to address pressing health challenges. And our mission is to connect academia, healthcare, the healthcare community, and the pharmaceutical industry. Our focus areas include oncology, chronic diseases, digital health, and health equity. Next slide, please. And our services are broad. We have a broad range of services that we provide to our clients, including grant writing and consulting for proposals. We also offer digital health solutions, partnering with our clients to develop apps or tools for healthcare, to support healthcare. We are also heavily involved in capacity building. So we create workshops, seminars to meet our clients' need for education, if they have need to train their staff or students on certain areas of interest. We also help our clients in research design and biostatistics. And we also partner with them to for their community engagement and efforts that they may have. And so I'll hand over to Dr. Adekipe to continue the rest of the presentation.
Thank you, Dr. Kano. We started Kotaraku, so we saw a gap, the need for healthcare research and consulting firm that doesn't just generate the data, but also translates it into solutions that are community-centered and scalable. We sit at the intersection of academia, like Dr. Kano said, research design, data analysis, publishing. We also support healthcare providers by helping them with patient-centered outcomes, survivorship programs. We are involved in community organizations, advisory boards, engagement, and also assisting in turning evidence into scalable, sustainable impacts. So what really makes us unique is how we approach this work. We have experience with evidence-driven, so bringing rigorous science to everything we do. And we're collaborative. We work across academia, healthcare systems, and industry. And we're also global. We've worked in the US, in Nigeria, for example, and I'm currently in Korea doing some work. So as you can see, our scope is quite wide. And here's just a way to highlight potential opportunities for Unilag audience and also people that are not from Unilag. We are able to expand opportunities to faculty and students, work on joint proposals for grants, access to mentorship and training programs, and also build evidence programs for Nigerian healthcare and public health. And I know that there's a lot of work that's already being done in Unilag, and this will just enrich it as a regional leader in research to practice translations. So here's how you can work with us. We're open to contract-based research, advisory partnerships, training collaborations, and also co-developing digital solutions. If you were in my previous talk that was just aired some minutes ago, I talked about an app we developed with my lab. So yeah, at Cotarex, we believe that the best way to transform health is to do it together. And thank you so much to Dr. Onwo Chuluba and Dr. Joseph for the opportunity to be a part of this. And here's our contact information if you'd like to reach us through emails, or we are also on LinkedIn as well.
Thank you so much. Thank you so much, Dr. Adedipe and Dr. Carlo, for that presentation on Costa Rica Solutions, a research and consulting firm committed to driving innovation, excellence in healthcare delivery. We have come to an exciting segment where the winners for the various poster sessions will be announced. And to do this, we call on Professor Abimbola Shoyomimo. But before then, we have music interlude, while we collate and finalize the scores. Thank you. This is the University of Lagos, the University of First Choice and the nation's pride. The conference has been stimulating, thought-provoking, engaging, and very informative. Do well to continue your collaboration and networking even after the conference. And please, for Costa Rica Solutions, the link that have been shared with us, kindly contact them and engage with them. Thank you. Thank you for it. Thank you for your patience. It's now time for the award ceremony, the best poster presenters, and award of the assessors. We call on Professor Abimbola Showemimo to make the presentations. Prof. Ma.
Thank you very much, distinguished ladies and gentlemen. We appreciate the time that you have spent with us. And so without much ado, I will go ahead to announce the best poster presentations, the 5 different segments that we have. We will start from pharmaceutics and pharmaceutical technology. The best poster presentation was born by the person with the abstract 020, and the title is Development of a tenofovir levonorgestrel vaginal multipurpose prevention technology, MPT film, using a quality by design QPD approach, in vitro release and in vivo safety evaluation. And the presentation was done by Mohammed R. Next, we have the presentation from pharmaceutical chemistry. And the title of that presentation was Identification of Potential Antidiabetic Ligand from Agemon mexicana leaf-derived compounds using molecular docking and pharmacophore modeling. And the presenter for that abstract was Oyawaluja B.O. Next, we have the one from pharmaceutical microbiology and biotechnology. And the best poster presentation was done from abstract number 028. And the title is Unveiling the Biosynthetic Landscape of Endophytic Streptomyces species, PGLA-C3X, via growth optimization and whole genome analysis. And the presenter was Ezibiora CE. For clinical pharmacy and biopharmacy, we had the best poster presentation from the abstract 033, and the title, Attitude, Perception, and Willingness to Take Hepatitis B virus vaccine by undergraduates in health-related courses in a tertiary institution, Lagos, Nigeria. And the presentation was done by Aji Laron. The last award for pharmacognosy goes to abstract 010, Olabode KO, and the title was Phytochemical Analysis and Inflammatory Activity of the boiled seed extracts of Plukenetia conifera, Unilag Ifobiaceae. Thank you very much.
Congratulations to all our winners. We congratulate the oral presenters and the winners of the poster presentations. It's now time for award to be presented to our assessors. We have 10 assessors, and to announce their names will be Professor Abimbola Showemimo. Is by Dr. Abimbola, Professor Abimbola Showemimo, Prof. Ma.
Thank you very much, Dr. Nkemehule. So for this conference, we had 10 assessors. We had 2 people each from different departments. So from pharmaceutics and pharmaceutical technology, we had Professor Ikora Hewo and Professor Oluyemisi Bamiro. We want to say a big thank you to the two of you. Also from pharmacognosy, we had Professor Kolawole Kayode Ajibeshin and Dr. Temitayo Ajayi. We say thank you very much to you. For clinical pharmacy and biopharmacy, we had Professor Gona Brian Oyebuchi, all the way from Ebonyi State, and Dr. Foluke Adenike Ayeni. We say thank you very much to you. Pharmaceutical microbiology and biotechnology, we had Professor Charles Okechukwu Esimone and Dr. Adeshola Olalekan. Thank you very much. For pharmaceutical chemistry, we had Professor Mbang A. Owolabi and Dr. Olusegun S. Ajala. We want to say thank you to all our assessors, and we assure you that your certificates will get to you very soon today. Thank you very much. We appreciate your time and energy. God bless you.
Thank you so much, Professor Shoemimo. So the assessors, your certificates are ready and you'll get your awards. Congratulations. It has been an interesting conference. Very stimulating, informative, educative, interesting, and impactful. A round of applause for yourselves, all conferees. Congratulations. Congratulations. It's now time for vote of thanks. And to give us the vote of thanks for this conference, we call on Professor Azubike. Professor Chukwuemeka Azubike is the vice chairman of this conference, and he has been interacting with participants, so you can see his face today.
Good afternoon, everyone. The Vice Chancellor of University of Lagos, the University of First Choice, members of University Management, the Dean, Faculty of Pharmacy, the keynote speaker, invited guests, colleagues, students, sponsors, members of the press, ladies and gentlemen. On behalf of the Faculty of Pharmacy, University of Lagos, and the entire conference planning committee, it is an honor to deliver the vote of thanks at the close of this stimulating conference, the 4th Faculty of Pharmacy Scientific Conference. We are very grateful to our Vice Chancellor for her unwavering support for academic excellence and for drive for a future-ready Unilag. So the Dean Faculty of Pharmacy, your support has been excellent. And in sustaining this conference series, we say thank you to all the past deans that will be there for us. Our keynote speaker, our eminent panel speakers from across the globe, you have actually broadened our views and stimulating presentations and invaluable insights. Special appreciation goes to the planning committee under the able leadership of Professor Margaret Sofedia, whose tireless efforts ensure the success of this two-day event. To our partners, our sponsors, and all who provided technical support, we say a heartfelt thank you. We sincerely thank our participants because without them, the conference will not be a reality. We thank our assessors, the practitioners, our students for your active engagement, thoughtful contributions, and shared commitment in advancing pharmacy and healthcare systems. It is our hope that the collaborations, the networks, and ideas directed here will continue to flourish beyond this conference, translating into impactful research and strong health outcomes for our communities. Finally, I thank the Almighty for granting us the grace for a successful program. We look forward to welcoming you all to our 5th edition, which we promise will be much better than this session. I say thank you, and God bless you, and see you all next year. Thank you very much.
Another round of applause for the Vice Chair, Professor Chukwuemeka Azubike, for that warm vote of thanks. Whatever has a beginning has an end. Thank you all for joining us for this year's conference. And like he said, it's an annual conference, and we expect you to join us next year. Please continue to interact with us on the WhatsApp platform in case you have questions and further communications. Your certificates are already in your email, and if you have not received your certificate by now, you're going to get it before evening. Thank you so much for this opportunity, and our prayer is this, that we continue to prosper, we continue to be in good health, and we continue to do our best in research and collaboration. That next year, when we gather, we will be better than this year. God will preserve our lives, and we'll see ourselves again next year. It was a great pleasure being your moderator for this 4th conference. I am Dr. Florence Nkemehule. Congratulations. Now we take the University of Lagos anthem, followed by the national anthem. Goodbye. Thank you and goodbye.